For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started. All right. Looks like we are live and ready to have some fun. I hope everybody appreciates the thumbnail as much as we do. Certainly represents the critics perfectly in the fact that the evidence that we are going to present tonight, the critics, the evolutionists, the scoffers, they do nothing but dodge and avoid that evidence as best as they can. Essentially just deny the reality of genetic entropy, genomic degeneration. So this is going to be a lot of fun. We've got Brother Ramat, Brother Will here. The reason why we're doing this show, titled The Destroyer of Fish-to-Fisherman Evolution, Genetic Entropy, the Evolutionist Worst Nightmare. It really is the Evolutionist Worst Nightmare. They've got no good rebuttal, no good argument against this reality. Okay, the fact that mutations are accumulating from generation to generation. Okay, far too quickly for selection, which is limited, far too quickly for selection to filter out all of these deleterious mutations. And we are going to be covering it in great detail, as well as refuting all the best arguments that the critics have to offer. So Will, he has been doing a study, a personal study on genetic entropy, from my understanding, and Will, correct me if I'm wrong, you just recently, you've at least been reading through genetic entropy, so therefore you got some questions, some input, you've learned a lot, so you've got a lot to say on this topic. So Will, good to have you. Matt, good to have you. Will, what are your thoughts in general of genetic entropy and your current study on it?
Well, mutation are accumulating We're going down. That's what the evidence show. We're looking at the evidence. The evidence show that, it's a recent creation that has been devolving, as God says, for thy sake I will curse thee. He cursed the ground, everything went down, it's lying down. We can see that from the, if we read into Genesis 8, I think we can see that the age is decreasing, everything's decreasing, the radiation, the magnetic field, so we're decreasing down, more mutation are accumulating. We're just lying down. There's a lot of papers on it, a lot of evidence from creationism, from evolutionists, from agnostic. So I think the evidence are really showing up that we're lying down, we're degenerating, not evolving. And I think it's a big problem for common ancestor. If we have been there for 2,000 years, it's not possible with the numbers of mutation we will have in our genome. So yeah, that's my thought on it.
Right. Good point. Good point. So basically, guys, in the chat, and then Matt, elaborate or give your thoughts if you want to add anything to this. But basically, we know now that the mutation rate is extremely high. Okay. It's roughly 100 new mutations per person per generation, and roughly 3 new mutations every cell division in our body. So it turns out that there's genetic entropy on a personal level, right? Generally, that's why we die, is due to mutation accumulation. But there's also genetic entropy on a population level, because many of these mutations, okay, are passed on to the next generation. And here's another thing that I want to point out too, is that harmful mutations, okay, vastly outweigh truly beneficial mutations. Okay, so the fact is, more harmful deleterious mutations, they are accumulating in the genomes of living organisms, okay, from generation to generation, with beneficial mutations, truly beneficial mutations, being incredibly rare. And that's the problem. Most of these mutations are deleterious. Natural selection is only limited to removing the worst, most detrimental of mutations, okay, because nobody disagrees with natural selection. And natural selection can amplify the best of the beneficial mutations, but it can do nothing against these nearly neutral, these effectively neutral mutations that are pouring into the genomes of living organisms generation after generation. Okay, so that's pretty well the basics of genetic entropy. We are all more mutant every single day. Okay. And there's really nothing that natural selection can do to stop the inevitable genetic degeneration of our species, of humans and other living organisms as well. It may be able to slow it down, right? But it can't actually stop this process. So Matt, thanks for being here, brother. Anything you wanted to kind of add to that, elaborate in terms of just the basics of genetic entropy and what it is?
Sure. Let me know when you're gonna make sound. I'm gonna mute up in a minute, but yeah, real quick. I'll share a screen real quick, and let's look at. This is what you just mentioned. I'm gonna enlarge it because. Wait, can everybody see? Oh, there it is. It's just lag. This is what you mentioned.
I just square. Yeah.
So it's the accumulation of 100 to 200 new mutations. The number can range. Sometimes it's down to, about 47. Sometimes it's around 230. But in general, this is the accumulation. Now, this is one aspect of genetic entropy. So some studies will tell you this. We define mutation rates as the rate of which the mtDNA type of an individual changes rather than the rate of mutation accumulation of an individual. So that's when you're talking about genetic differences, mtDNA differences between people. So sometimes what I'm getting at is you will find a mutation rate study will sometimes look at accumulation, but for the most part, they're going to look at differences, right? They're going to be trying to say, okay, how many mutations are there that have accumulated that are detrimental? How many can we count, and how different are we from one another based on that criteria? So we are seeing the accumulation of 100, but those are near neutral. So they're also affecting us. That's what John Sanford has discovered as well, because they're not neutral, they're slightly deleterious, but selection cannot see them. And for those that say, well, you know, selection can see them, well, some bad news on that front is they have, sorry, you gotta do everything for me tonight. You gotta share my screen again. This was their comment. Selection is unlikely to be a major factor that underlies the differences between the phylogenetic, which is the evolutionary, and the pedigree divergence rates, which is not the same thing as saying that selection does not act on mtDNA. So they're saying that selection can remove them, but overall, it doesn't have that much of an effect. It can't do as much as they're saying it can do. And they still can't figure out the disparity between why the phylogeny says it's here and the pedigree, the observed mutation rate, is saying it's so quick. So there's that disconnect. They're saying it's selection, but selection can't do it. So they're admitting it. And this is what, sorry, I'm going to share screen. I'm going to keep you busy tonight. This is what
That's what I'm here for.
This is what people think of with genetic entropy right here. Oh, right there. That's genetic entropy in action, guys. That's more inbreeding, actually, but still, it's another example. That's down, not up. How is that guy going to evolve into anything in the future? He's not. He's going down.
You have to give him time.
Right. And that's the major problem. For one, inbreeding, as we always say, is a sneak preview into where we are going genetically as a species, right? It's like a small scale example of genetic entropy, okay, as the human population accumulates more and more mutations from generation to generation. But when it comes to the resolutions or so-called solutions that the evolutionists are putting forth, they all fail essentially, but a lot of them are misrepresentations, as in they make it out to sound like creationists or proponents of genetic entropy ignore the role of natural selection, right? No, we agree natural selection happens. The problem is it is limited. Okay, as you were pointing out, these effectively neutral mutations that are only slightly deleterious, right? They are pouring into our genetics from generation to generation. And because they're effectively neutral, they're only subject to what? They're subject to genetic drift. Okay, they're essentially invisible to natural selection. And that's why the bigger picture of this all is the fact that the evolutionist explains the origin of all genetic diversity as the result of mutations over time. Okay, these typographical errors in a text, right? So that's why they're going to have that problem, is they want to drive their fish-to-fisherman through a process of genetic mistakes, deleterious mutations that are eroding genetic information essentially from generation to generation. While we as creationists, we recognize the fact that mutations are the destroyer and not the creator. So we explain the vast majority of DNA differences as the result of what? De novo creation, created heterozygosity, as James Downard loves so much. So that's the major problem with their models. They want to explain all these DNA differences as the result of mutations. No, now we know mutations are incredibly damaging, incredibly deleterious, and most of them being unselectable, invisible to selection, okay, because they're effectively neutral. They're only subject to genetic drift. My question to you, Matt, is you'll hear the evolutionists, they'll acknowledge the fact that mutations are accumulating, right? They want to assume most of them are absolutely neutral, but they'll say that there's no actual observable evidence, empirical evidence, that genetic entropy is occurring in populations today. What are your thoughts on that? There's no evidence for genetic entropy. It's all made up. It's not real. Young Earth creationists invented it. What are your thoughts on that?
Well, if we made it up, how come all of these people right here acknowledge it? How come Kimura, called Kimura's Quandary, right here? You know, we have Mueller's ratchet, and we have all of these examples of people that have come across this problem and said, interesting, what are we going to do about it? and how can we solve the problem, because it's a problem with evolution. And then we get people on the internet that come and say, none of these people are real, I guess. I honestly don't know what they say. They probably get mad that one of these is a creationist, so therefore none of these people can be correct. That's probably their best argument, because it's a reality. And it's just what we see. It's what we know. I mean, what do we know? Beneficial mutations are extremely rare. They admit that. They're hard to prove, and they're very, very rare. So then we get empty DNA. What do we see when we look inside the mitochondria? We find, wow, it's surprisingly common to have mutations arise. Well, that shouldn't be, right? If these mutation rates are supposed to be slow and we're not supposed to be seeing them, then how come they're fast? How come we see them? How come when we look and, oh, look, there's genetic meltdown of gorillas. Why? Because of just like the Neanderthal, isolated and exactly what we expect. How about, actually, we'll go right here. How about the mammoth? What happened to Neanderthal? The same thing that happened to the mammoth. They migrated north. They got stuck in the snow. They had genetic meltdown, fast mutation rates. It killed them, just like it killed Neanderthal. It's not a myth. It's not some mystery. We have the evidence. How about some animals alive today? Where do we get mutation rates from, right? Remember, I said earlier right here that mutation rates, it's not about accumulation. It's the rate of change. So how come when we look at modern day chickens, what do we find? We find exactly what we would expect looking at humans. The mutation rate right here shows that the rate of evolution in the pedigree is 15 times faster than it's supposed to be. Based on what? Evolution. They base their predictions on the assumption of evolution being true. So they go by the phylogeny rate, which is a split. This is the human split. And it's based on calibration. It's all based off the fossil record. So because the fossil record isn't true, therefore their mutation rates aren't true. It's really that simple. And they even admit it. Unfortunately for them, look, they even say it in their own words. When they look around, they go, there is no trace in the geologic record of any global event 200,000 years ago. But we have the genetic evidence that tells us there was a genetic reset in all life at exactly the same time. So literally, we have genetic evidence. They went to the physical evidence and they said, wow, our made-up record isn't true, but the genetic evidence is. So we're just going to go with the genetic evidence and say we don't know what in the world caused it, but it happened anyway. As far as the evidence, I guess we'll figure it out one day. So they don't really care because they know they had to retrofit the data no matter what, because the genetic evidence doesn't lie. And it's not just human beings, and it's not just like chipmunks and squirrels were genetically reset. It's everything in life, even the aquatic life. So a volcano couldn't do it, so they can't blame it on the Toba catastrophe, like one massive volcano destroying all life. They can't blame that because that sure wouldn't hurt a sea urchin on the bottom of the Mediterranean Sea. So they were like, okay, we have a problem. So that in the meantime, they're just going to deal with it and say, we'll figure it out eventually. But they stick to the 200,000 year date because that was one of their early on predictions. So they have some problems with that. So I like to point out that it's an all species problem, right? Most species are not driven to extinction before genetic factors impact them. Well, that's pretty odd, right? So if they're going extinct, what would you expect to have happen? Well, exactly what we say is happening. They're going from a heterozygous state into more of a homozygous state. Mutations are accumulating. They might be being inbred. It might be even faster. And then they die. Pretty simple. And then here, right from their own mouth, multiple different places. What happens? Now, these aren't just mutations. These aren't the de novo near neutral mutations that always get handed down. These are harmful mutations. Look at that. Exactly what we would expect around 5,000 years ago. An explosion, about 200 to 400 generations, harmful mutations just came out of nowhere. They just exploded on the scene, and here they are. Detrimental mutations everywhere. That's not what evolution should be saying with mutations. These mutations are what need to be there to cause diversity. And they're admitting that these mutations arose thousands of years ago, a few hundred generations ago. So which is it? Would the 200,000-year-old bottleneck cause all the diversity and what we're seeing? Or are you admitting that what's really causing diversity only arose just a few thousand years ago? You can't have your cake and eat it too, right? And that's just,
there was a paper about like the E. coli when they were speaking about like they were breaking down a bunch of disease, like they did like a bunch of generation of bacteria or something like that, and they were speaking about how they break out apart, like trying to survive. Like, this is not what evolution is supposed to teach. Like, it's supposed to gain stuff in order to get where it is. And how is it that, in order to survive, it lose a bunch of information in the genome, like it's split apart? I mean, how does evolutionists explain all those papers? There's literally papers on, I think it's like on Nature or something like that, where we're speaking about like the disease into the E. coli. It's like the O157, something like that. It was like breaking down apart. They were literally saying it was gonna go extinct, like it was gonna die off, like the whole generation. I mean, that's a huge problem for them. It doesn't explain how it get there. How did the information get into the genome? Only what we see is that the mutation are breaking apart. The genome is breaking apart in order to survive. It doesn't explain how it got there, and they cannot explain that. And B, he wrote an article about that too, which is pretty good.
Yeah, they don't like the origin story too much. They would rather just start talking about the game after the dominoes are already standing. They don't care where the dominoes came from. As long as they're standing, that's all they care about. That's all evolution is, right? Remember, abiogenesis, don't touch it, don't deal with it. That's their motto.
Yeah, exactly.
That's exactly right. And we know it's so much easier to break something down than to actually build it up, right? And it's so funny how they'll say that there is no evidence for genetic entropy, genomic degeneration. It's a made-up concept by creationists. And as you pointed out, Matt, the fact that most deleterious mutations, as I put up here, the fact that they have extremely small negative effects on fitness, they're invisible to selection, they build up, they're subject to genetic drift, and they eventually degenerate, right? This has been acknowledged by non-young earth creationist scientists, right? I mean, paper upon paper here. Slightly deleterious mutations. Kimura acknowledged it, of course. Lynch. Mutational meltdown, for example. Mutation accumulation and the extinction of small populations. Quantifying the genomic decay paradox due to Muller's ratchet in human mitochondrial DNA. So this is recognized, and what they do is they invent artificially contrived rescue mechanisms like synergistic epistasis, mutation count mechanism, okay? Or they will point out that humans, for the most part, selection's been removed, okay? Because now we take care of our sick, right? We help each other. As compared to in the wild, a lot of these mutations that may thrive in the human population, in the wild, okay, that mutation may be removed by natural selection, which means the human population may degenerate faster, okay, than say some of your populations in the wild, because selection can still help to slow down the process, but it's not going to stop. Natural selection is not going to stop genetic degeneration even in the wild, because the fact is the effectively neutral mutations that are not your big, massive mutations, okay, they're still invisible to selection. And the one thing that I wanted to point out too, I'm going to stop sharing my screen here, is this argument that mutation can solve the problem, as in once enough of these deleterious mutations build up, now Mother Nature will be able to see the damage, for example, and remove the mutated. But the problem is even if selection were to remove the worst mutants in the population, the fact is we are all still mutant, right? We are more mutant than our parents and more mutant than our grandparents, okay? So you can remove the worst, but you're still stuck. You're still left with people who are more mutant than the generation before it. So that's why it is inevitable that mutations will accumulate. So this whole mutation count mechanism, okay, for one, it's not biologically real. The model doesn't even remotely resemble reality. And all of these artificially contrived mechanisms, they've been dealt with in numerical simulations. They've been shown to fail especially whenever realistic scenarios and realistic mechanisms pertaining to selection, purifying selection, are integrated into these simulations, it still shows that genetic degeneration is inevitable. And here's what's funny. I'll say this and then I'll yield. Because Kimura, right, he just threw it out there. You know, beneficial mutations will counterbalance the damage, but he didn't do anything to demonstrate that. Okay, people like Dr. John Sanford, they've looked at this the most. They've looked at beneficial mutations. We know they are rare. And even your rare beneficial mutation is still deleterious for the most part, right? So it's about 1 in a million. And there's still typically deleterious effects associated with that mutation. So therefore, okay, for sake of argument, evolutionists, let's give you 1, let's give you 10 beneficial mutations that are truly not deleterious. That's still not going to counterbalance the damage due to the constant influx of these low impact deleterious mutations. A few rare beneficials, give it to them. It's not going to counterbalance the damage. What are your thoughts, gentlemen?
Yeah, go ahead, Matt.
Oh, I was just saying, yeah, how could they? How could they? I mean, you're literally, they're trying to say that selection is going to remove these deleterious mutations. Well, wouldn't that essentially get rid of diversity in life? Wouldn't we have almost no diversity if it's weeding out mutations as they're arising? It's literally countering their very statement, right? And what do we know when we look in the mitochondria? There's a saturation point that needs to be reached before you no longer can see a mutation, because it eventually starts turning over the same allele. Dr. Jensen ran it and said, you know, look at this. We have about 1,000 different changes going on. Here, I'll share it real quick. I'll show you what I mean. Right here.
I got it.
Thanks. So you can see Dr. Jensen's chart right here, and there's 1,483 actual mutations that have occurred over the amount of time, right? So if evolution were true, we should see about 12,000 mutation saturation point, but we don't see anywhere near that. You know, if evolution was true, there would be about 21,457, right? But of course it would be maxed out at about 12,000. We don't see anywhere near that, nowhere. So what's going on? We see genetic diversity inclining, you know, going up quickly, really, really fast, really rapid with fast mutation rates, but we don't see saturation whatsoever. That would have easily been reached in the evolutionary time scale. So something's going on. What is it? What's going on? Well, the bottleneck wasn't that long ago. That's it. End of story. That's simple. We don't have to invoke the problem of, well, maybe the mutation rates are different and selection is removing them. You know, that's just a rescue device. We see them occurring. We don't see selection removing anything but the worst. So to put a spin on it is just a rescue device. That's why we call it that. That's why I wrote a book called The Rescue Device. That's all it is. They just keep reaching.
Yeah, Matt, you are ignoring variation in fitness, and you are ignoring equilibrium of beneficial mutations and negative mutations. So, you know, you're just wrong, because humans today exist at an equilibrium, right? There's a mutation / selection balance. So how do you respond to that?
Well, if there was, then what's going on between the discrepancy between the African and the European lineages, right? We see the Africans have the highest, and then we see Europeans with the least amount. And evolutionists come along and they say, well, that's because Africans have been around longer. And then Dr. Jensen comes along and says, real quick, no, it's because they have a faster generation time, and that would speed up mutation rates. And therefore, they would have more differences because more generations have occurred. And then they might come along, if they're smart, and they would say, well, wait a minute, if Africans have had more generations, then wouldn't that mean genetic entropy would affect them more? Now, it would, except for other races were interbreeding early on, Europeans especially. So we broke our PRDM9, so recombination rates don't happen as quickly. And then all of a sudden we have more genetic entropy in these founding parents, and then we branch out, and now we have genetic problems that come along with that problem. That's why we have more genetic problems than the Africans do, who have had more generations in that time. So right. It's so easily answered in our model.
And Matt, people like Dan, they'll say, as long as you have selection, you will reach a selection / mutation balance through this selectable variation within relative fitness, right, in populations. So Dan is saying that we will never actually go extinct because the most mutant will still always be selected out of the population. So he'll say because of that, there is no reality of genetic degeneration. How would you respond to that?
Okay, well, if this were the case, then we wouldn't see the problem of health that we actually have today in today's people. You know, we are literally having the first generation of people being born where their parents are outliving their children. We have heart disease, diabetes, cancer rates are off the charts. Almost 1 in every 2 people have them. Not to mention we have fungus, mold, yeast, bacteria, viruses, parasites, heavy metal toxicity, free radical damage, acrylamides, mycotoxin. We can go down the list of all the different things that are corroding the body away and bombarding us. And we're getting genetically more broken down all the time. We have the average human being that's been tested is missing 100 genes. Those aren't just genes that are broken over time. They're completely missing now. They're like destroyed. We don't even have them to pass on, right? So there was a gene that your ancestors had that broke so badly there was actually nothing to pass down and for the grandkids to inherit. We have a gene like the MTHFR that's a notorious methylator. It determines on how well our bodies are going to methylate and cleanse out. And it also determines on how well functioning other genes are. This gene is broken by about 50% in the average person. And that's just if one mutation is in the gene. If there's 2, it's broken 70%. And that controls the functionality of the genome. So we have that. I mean, if this doesn't tell you something
Matt,
right.
Now, if I were to grant you everything that you're saying, okay, diseases accumulating every single year and every single generation in human populations, the fact that humans are degenerating. But what if I said this is simply because natural selection has been relaxed in the human population? Therefore, these diseases are allowed to accumulate, where in the wild, right? In the wild, these diseases would not accumulate because there is not relaxed selection in the wild. These mutants in the wild will be selected out of the population, and therefore those diseases and those mutants will not thrive as they do in the human population. How would you respond to that?
I would have to say, well, that's easy. We have animals that nothing's like a human, right? So we have the ability to survive genetic entropy because of our massive amounts of rapid population growth and our dispersion. We migrate, we travel extremely fast. We're like birds, right? We migrate, we travel around the world, and we can live in every diverse environment that there is. So we're able to withstand this genetic entropy. That's why I said earlier, when we start looking at the places like the gorilla, for example, right? They're not migrating around. So their population size starts to dwindle, and they're stuck in the rainforest in Africa. And guess what? Genomic meltdown. Exactly what would have happened to us if we were not migrating and traveling around doing that. So just because we don't see that in the human beings as much as we see it in the animal kingdom doesn't mean that it's not happening to us. We do see it in the animal kingdom. And
Well, real quick, I'm saying that we do see the thousands and thousands of new diseases in the genetic database in humans every single year. But I'm being devil's advocate here. I'm saying that the critics will say that is only because there has been relaxed selection in the human population, right? We take care of the sick, where in the wild, the lions or the zebras or these various animal species, they're not taking care of the sick. So therefore, there is no relaxed selection in the wild. So the only reason we're seeing incredible genetic meltdowns in terms of diseases, the critics will say that's only because there is relaxed selection in the human population today.
Of course they can invoke that all they want, but it's happening in animals as well. We look at the we're looking
right. So that would be the argument. Is it happening in animals? Would be the argument then. Exactly.
We look at a polar
bear. You're
bear.
saying yes, it is, right?
Of course, yeah. We look at polar bears; they're in genetic meltdown, right? And they're migrating, they're traveling as much as they can. They're blaming global warming. It can be blamed on anything, but I'm just saying that they're going through this. They have 15 genetic diseases on average, and they're being really hindered really, really hindered from this. So they would need to do what we're doing, and they would need to explode in population size, and they need to diversify and stop inbreeding, right? Because they're becoming more isolated and they're coming down. They're going to die. That's it. So the only way to survive genetic entropy is through high population growth and migration. You have to disperse, and then you don't see genetic entropy as fast. So the fact that we see it in humans at all, it shows you how detrimental genetic entropy is. And think about this. If genetic entropy wasn't a problem, and why wouldn't they be just using the evolutionary model when it comes to health? This is the National Institute of Health, and they use the creationist model. If they don't care about creation or evolution debate. They don't care which one is right. They don't care about the internet bickering that goes on. They care about what makes people better, what cures you. Well, why are they using the creationist model? Because that's the reality, and that's what we see. We see it with dogs because we've been breeding them, and then they go, oh, that's because they're out of selection. Ah, well, go look at wolves and tell me what you see there. You'll see the exact same thing.
Right.
Great stuff.
So, and here's the thing. The reality of genomic degeneration, it puts shelf lives on genomes. And that's the point, is the species that you see in the wild that have not yet gone extinct due to genetic degeneration. It's because they have not been around for as long as the uniformitarian and the evolutionist would have us believe, okay? There are shelf lives on the genomes of living organisms. And the problem with the critic is they have a rescue device for all of these instances that you're pointing to, right? For example, polar bears, they'll say it's due to climate control. When it comes to the specific case of genetic meltdown in gorillas, they'll say this is because it's a small population. They're subject to inbreeding, right? They'll say the same thing with Neanderthal populations, mammoth populations, butterfly populations. We have all of these examples of genetic meltdown in these small populations, and they want to say that it is because they are subject to inbreeding, right, as you were pointing out. But then we go all the way back to what we were talking about at the beginning. Inbreeding is a sneak preview into where we are going genetically as a species. Yeah, humans have a much larger population, but it doesn't change the fact that these mutations that are accumulating, the effectively neutral mutations that are accumulating from generation to generation, they're still only subject to genetic drift, okay? So even if these arguments, okay, even if we allow for these arguments that Dan is putting forth, that I was kind of playing devil's advocate with you on in terms of relative fitness, okay? Even if there is relative fitness, okay, and no one's denying this, say between individuals within the population, and the fact that some individuals are better than others, it doesn't matter because we are still all multiply mutant. That's why I pointed out you can get rid of the worst, but you're still left with people who are more mutant than the generations before it. Okay, so that's the problem. There can be selectable variation within relative fitness, okay? But populations still only get worse. This is natural, okay? This is continuous because the mutations are continuously accumulating. You can go as far as this. You can go as far as this. Take the human population today, let's say 8 billion people, okay? The fact that each generation has more mutations than the generation before it. Remove 50% of the population that are the most mutant. Just remove them, okay? What we are left with is now 50% of the 8 billion people that are still more mutant. Okay, so all of these arguments, equilibrium of beneficial mutations and negative mutations that Dan looks to. We can look to fitness variation as well. Once again, all of these mechanisms only do what? They only slow down the genetic degeneration. They don't solve it. And yes, these examples of mammoths and polar bears, okay, these examples of butterfly populations, these examples of genetic meltdown, it's a sneak preview into where we are going genetically as a species, because inbreeding, right, reveals the hidden reservoir of genetic mistakes in the genome and leads to rapid and accelerated genetic degeneration, which is where we are going as a human species. So even if there is some relaxed selection in the human population, all that's going to do is make the problem worse for human populations and a little bit better for animal species. But the effectively neutral mutations are still invisible to selection, just as much as they are in humans and just as much as they are in animals of every kind. Go ahead, gentlemen, if you guys had a couple points you wanted to make there.
Yeah, I correct me if I'm wrong, but isn't natural selection more of a filter than like removing the mutation? Doesn't it just filter them?
Well, you can think of
Right.
selection, being able to see, like, for example, if an A allele is beneficial, like let's say that there's an SNP that is homozygous, and it's in the father, but he marries somebody where it's heterozygous and they don't have a G or an A, and a T comes along and it's a harmful mutation and it gets pushed into the child, but it's really, really harmful. What'll happen is selection can just remove it. It can see it. It'll be like, you know what? If I don't get rid of this, it's going to die. And
Yeah.
selection can pick out these really detrimental ones that your life is in danger for. But the really small, the tentative ones that it doesn't care about, it doesn't even care to remove them because that's not its purpose, right? It's just trying to save you. It's basically your rescue device.
Yeah, and like let's say like even if we remove off of the population, I mean there's still more bad mutation than good mutation, so it's a big problem for them.
Yeah.
You know what I mean?
Yeah, well, that's the real big problem with inbreeding, right? Because once both parents have a mutation and they become homozygous and they both have these detrimental mutations, the offspring stands no chance of ever being able to have selection remove them. So it's a guaranteed bad mutation every generation. And the more you inbreed, the more generations happen, the more of a problem you get, until eventually it can't. The compounded mutation load is too much for the species to handle, and you're just dead. So just like the mammoth, they couldn't take it anymore, right? They were up in the Arctic in the north, and it was really cold and harsh and barren, and they were just small groups scattered around, just like Neanderthal, right? And pretty soon isolated. And then pretty soon they were like, we need to get out of here probably. So they started migrating around, but they stayed in the tribes because you need each other in harsh conditions, right? And then inbreeding kept happening. More mutations kept pouring in. Boom, gone. That was the end of them.
There's too much accumulation. Like the good mutation cannot override the bad mutation because they're extremely rare. Like you point out, from those paper, they're super rare. The good mutation are rare, but the slightly bad mutation are more common, 60 to 200 mutation per generation. So let's say let's just give them that. If you get one good mutation, one beneficial mutation every generation, but you get to 60 to 200 mutation that are slightly not good, how long does it take to overtake the bad mutation by good mutation? It just doesn't happen. If you get one every generation and 200 bad mutation every generation, well, it's never going to overgrow the other mutations. So that's a big problem for them there because there's too much bad mutation and not enough good mutation to survive. So that's why they end up dying.
So that's a great point. And this is one of their go-to rescue mechanisms, okay? Kind of what you're touching on there. I debated Dr. Stefan Frello over a year ago now, actually. And his main argument for genetic entropy, to counter genetic entropy, was the argument from there being a trade-off, right? These beneficial mutations, okay, sometimes they'll look to what's called a super beneficial mutation, will counterbalance the damage, right? But the problem is, because of the influx of all of these massive numbers of low impact deleterious mutations from generation to generation, those very few, okay, beneficial mutations that occur are not going to be capable of counterbalancing this damage. Look at it this way, okay? We've got a genome that is degenerating in many ways, okay? And then you may have a few single nucleotide sites, okay, based on the rare beneficial mutations, okay, that are improving, Let's say there's a few nucleotide sites that are improving. Well, from generation to generation, we've got massive numbers of effectively neutral deleterious mutations accumulating. That means we have huge numbers of nucleotide sites that are degrading. Okay, information is being lost. So that means this trade-off that they look to, okay, it's not sustainable, because do you know what a trade-off results in? A trade-off results in a shrinking functional genome size. That's actually what we see in Lenski's experiment right now. Okay, so basically you're just throwing out all this information, okay, from a multitude of nucleotide sites, and then the evolutionists want to try and replace all that information with, desirable point mutations or the rare beneficial gene duplication. Okay, it's not going to work. For one, most beneficial mutations are reductive and functionally compromising to the organism. Okay, so even your best beneficial mutations are not actually taking things forward. They themselves are degrading. I'm going to use this analogy and then I'll yield because I like this analogy. It's kind of like take a car and you want better gas mileage for your car just temporarily, okay? So you decide to start throwing things off, okay? Removing weight, remove the doors, okay? Remove the mirrors, take off weight. You're going to get better gas mileage for the time being. But guess what? Overall, this is damaging and degrading to the car, okay? It's only short-term advantage, but long-term damage. And that's what we oftentimes see in these evolutionary experiments, like the Lenski experiment. A lot of these bacteria, they are adapting for short-term purposes, short-term gain, but it's long-term degeneration. It's called adaptive degeneration. It's reductive evolution, okay? And this is the best that that they can look to. This whole trade-off or beneficial mutations, these rescue mechanisms like synergistic epistasis, mutation count mechanism, none of these rescue devices and arguments can solve this problem. Go ahead, gentlemen.
Yeah, exactly. It's like you have a car, you have some gas. Let's say you have like 60% of gas in, and now you're driving, and you hit something, it's gonna give you a bit more gas. But after that short period, you're going to lose more. They're just going to try to survive for that moment. But after, they're just going to degenerate more. They're just going to lose more and more and more since of that, boost. So that's how I see like mutation. Like, they'll just adapt by throwing off gene, by throwing off stuff. But after, it's going to big impact them because there's going to be too much of that stuff that has been lost. So it's going to be a big problem for them. In like, let's say 1 billion years, just so they cannot explain all those type of stuff. Like, they always claim that mutation is the mechanism that the whole diversity and complexion in our body, but all we see, it doesn't explain how it got there. It just explains how it survived by dropping off other gene, but they'll just dodge, say, well, just give it time. There's going to be good mutation that's gonna come off, and just remove those. But it's just dodge and dodge and dodge over and over again.
Well, it's one of the reasons.
Right. So you've even. Go ahead. Yeah, go ahead, Matt. Go ahead.
I was going to say that's why they are realizing when they look at these deleterious mutations on how harmful they are. They go, well, they obviously couldn't have been around that long because they're too damaging and we would have never survived. That's why they threw them back 5,000 years. Now they can account for the reasons, like, oh, it was probably farming when we started industrializing our food, and then we started eating grain that we've never consumed before, and it started mutating the body in a way that it's never done. Or maybe something happened to the atmosphere and there was a big change, and this allowed more mutations to really start bombarding it. And then we messed up the ozone layer, and then the ozone layer wasn't able to reflect a lot of the ultraviolet C rays that are the worst type of radiation, and that bombarded us, made it even worse. So there's all these reasons why they're accounting for deleterious mutations being more recent, but in reality, they've always been this bad. It's just not going back 200,000 years. They don't recognize what they're saying. You see, they know mutations are recent. They just can't make sense of it in the evolutionary view. So they come up with these ideas, right? These concepts.
These imagination.
Right.
Imagination, yeah. And
Well,
they
they
were just like, exactly. It's like, Michael B. He was doing some good stuff about, like, he has a good series on mutation and the big problem with it. He always goes around. He gave like the example of the polar bear. He had like a gene that was coding for his it was coding for his color, his skin color, and it migrated to the south. It went into a place where it was hotter, less wind and less like snow. And like literally when he entered there, like in that zone, like it took a couple months, and he literally break down his gene for his color coding. I don't know if you ever saw that. He did a paper about that, about like the result of mutation. So
no,
like it just keep dropping off. I mean, it doesn't explain once again how did the color gene get there? How did it forms? How did mutation lead it to, getting that color if it
Right.
just keep removing it over and over again? I mean, it just mind blowing just in my face. It's a recent creation that has been created and it's just dropping off. It's like it's just so obvious, man. How can you not see it?
it's
Right.
the same. It's the same thing with what they say about human skin color, right? Like, where did we get our skin color? Well, they said the first humans had black skin, right? That humans branched off and we evolved black skin in Africa because that's the most protective skin. And then over time, mutations came along and they turned our skin white when we started migrating and traveling around. It needed to, right? We're up in the cold north. We don't need black skin anymore because we need to absorb more vitamin D from the sun, so we need to be more pale for that. That's why women worldwide are always more pale than men, because they need to be, because they need to produce milk with more vitamin D. So what they do, they looked over and then they found primates are actually all white when you shave them. So they're like, oh, whoops. Okay, well, maybe a mutation arose to cause black skin first. And then once they got black skin, then they got the mutation that messed that melanin up to begin with, right? Because apes don't have melanin. They don't have any skin color. You shave them down, they're completely pale. They don't have anything. So they had to account for maybe a mutation came along, a new gene, popped up, and then a mutation arose, causing pigmentation to begin with in the first place. And then a mutation changed that and allowed other skin colors to evolve. Ours is real simple. Our models, it was created. You have it, you have brown skin, and it can code for all the different skin colors, and it can do it very, very quickly. What do we see? Exactly that. And guess what? You take two white people, they can, through recombination alone, they can never produce a black child, just like two black people through recombination alone can never make a white child. But yet a brown skinned person with recombination can do both of those things. So very unique, right? What does our model say? It uses gene conversion recombination as the major mechanism, and that's what we see. So just go with the evidence.
Yeah.
Actually, if I could jump in here real quick. Actually, first I want to point out that in the audience, C Science Film Labs. Good to see you, brother. Look up Young Earth Creation. I'm going to add, Matt, I'm going to add your channel where you are uploading all our videos and work. I'm going to add it in my featured channel section. And I wanted to point out that channel is over 5,000 subscribers now. And the flood video we put out like over a year ago is now at like 300,000 subscribers. So share that around. It's completely gone.
Oh yeah, man, that was amazing.
I mean, yeah. But I don't want to derail the conversation too much. So I just saw that, but I want to ask Matt because he's really knowledgeable on this subject. Even a lot of your so-called beneficial mutations that they look to and they try and argue for, right? Yeah. Those ones that are rare, that they say are going to counterbalance the damage. We now know that a lot of those are just general adaptation, epigenetic, for example, based on the environment. Can you touch on that a little bit, Matt?
Yeah, epigenetics is really bizarre because two identical twins can be born and yet they can move away from each other. One can move into the north and one can move into the south, or one can live in the high altitudes and one can stay low. And the genes on how they react to those environments will be totally different. So the person that's living at the high altitude will be able to be able to store more oxygen. So when they travel in those areas where there's less oxygen, they survive better. That's what the Sherpa people do. And they thought this was a beneficial mutation for a very long time because they said, wow, these people live up there. They must have evolved the ability to live at these very high regions to do that. Well, the problem is their offspring didn't have the ability. So if it was a mutation and it never got handed down, then what's really going on? Because lactose tolerance, you can hand down, right? So they're like, what's going on here? Like, what's really going on? So they figured out, no, this is just a switch. They live up in this region. The body says, I need to adapt to this. And it's just a flip that gets switched on. And you can control this yourself. I do it all the time. For example, I take tinctures for it. Now, as you can see from my face, I like fighting. So I'm getting hurt all the time. So how do I offset a lot of the damage that's being inflicted onto my body all the time? Well, I need to activate genes that are turned on that I need to turn off, because by turning them off, they turn on other genes. So for example, there's a process that's called mTOR. And mTOR, once it's turned off, activates longevity genes. And it's done by caloric restriction. Because you see what happens when we're deprived of nutrients, our ability to survive that relies based on our genes. So these epigenetic switches have to get flipped on or we're going to starve to death. So how does it keep you alive through a famine? That's epigenetics. So we've learned that genes are all based on these little tiny switches that get flipped on based on what we're doing and where we live. And none of them have to do with these mutations whatsoever. So everything that they used to believe were beneficial mutations have just turned out to be little epigenetic switches that we can turn on. And after about 3 or 4 generations, they're gone. They're no longer there.
Yeah. There's even a paper. It was published by his name is Alexei Kondrashvov. It's a Russian name. He says in modern human population, mutation with small eventual effect are probably accumulating faster than they are being eliminated by selection. Basically, he's saying that there's more mutation that are getting into ourselves than that natural selection can see.
Oh man,
but
he's lucky he's not on Team Dodgeball. He would have never admitted that one.
Oh yeah, but only John Sanford believe in genetic entropy is stupid creationists. That's just all they say. I mean, it's just apparently, according to them, we don't understand evolution, therefore we're not required. It's like Professor Dave saying that Dr. James Storr should not even be teaching science because he believed the resurrection of Christ. I mean, how stupid.
It just shows their incredible bias.
Yeah.
Yeah, you guys always heard that.
What Matt was saying, I just want to point out. Oh, I'm sorry. What's that, Will?
No, go ahead.
I was just going to say, so based on what you were saying, Matt, when it comes to these epigenetic related adaptations, right? So now a lot of their so-called beneficial mutations are just general adaptive episodes based on the pre-programmed capacity to change, right? These switches are already in the genome, just waiting to be turned on due to epigenetic regulation and the correct environmental input. So that's not going to help them. That's not going to help counterbalancing the damage. And now the few rare so-called beneficial mutations that are actually, let's say, mutation related, typically they are reductive and functionally compromising to the organism, which means there's nothing that they can look to that's going to counterbalance the damage. The fact is, what we see is reduction in genetic functionality. We see most mutations are effectively neutral. We see general adaptive episodes, let's say these epigenetic changes, which is perfectly consistent and expected from the design diversity starting point. So there's nothing that's going to save them. And as Will pointed out, I mean, you've got population geneticists that are recognizing, like Alexei Kondrashov, that are recognizing that man is presently degenerating. I've got brother John Maddox here. I'm going to bring him in. He said he had something to say. John Maddox in the house. What's going on, brother? What's going on? The floor is yours, man.
Oh, no, it's good. Say a few things about what you guys are talking about right now. I was backstage for so long, I forgot what I was going to say, though.
Was it about prophets or moron, Dave?
We're in detention, John.
I know, right? What was I gonna say? Well, in line with what you were talking about there on the passed down epigenetic tags for different things like, obesity in terms of starvation during or low nutrition during, I think it's the late stage of pregnancy versus early stage. It gets that detailed in terms of like where the tagging happens. Like if it's if the nutrition it was deprived like early, like the first trimester, for example, but then it got better before like the baby was actually born, then oftentimes the metabolism issues don't come into play. It's very interesting stuff. But in the even beyond that, I think the one of the points that really needs to be made about some of those epigenetic tagging things is like in terms of the recombination that's happening, at fertilization, they now know and have been able to observe the fact that tags are almost all of the uh, epigenetic tags are removed usually, right? Almost always removed usually. But then there's other ones that are kept specifically, and there's other tags that specifically delineate which portions of the DNA are from the mother and which are from the father. And it's like one of the reasons that they are unable to take like 2 male or 2 female DNA strands and be able to use them to create a new life form.
Mm.
It literally will not process. You have to have the tags, which they don't know where they all are, but they know that there's ones that are specifically female and specifically male. And if you don't have them, you're toast. You have no offspring. Period. And to me, when you really think about that premise, it creates a whole new level of conundrum for the evolutionary model of this means you have to have all the mechanisms. I'm talking about on the protein level, right? To be able to do the tagging and untagging. We now know that there are specific binding site regions in the DNA itself, which are what helps to control or to recruit different methylations, right? And then you have to have the actual read, write, and erase proteins to actually go and do the functional execution of those tags. Right? I mean, to me, when you contemplate all of that, I mean, it's hard to even comprehend. When you really think about that, at least for me anyway, when you think about like this is all happening on the molecular level with theoretically no conscious activity, it's just done. But it's done in real time based on external variables that are the result of signals being sent oftentimes from extracellular locations through receptors on the surface on the membranes, and then sent into the chromatin to be able to then to execute all these other functions. I mean, it's so far above our level of computational capacity right now. I'm talking about for both, I think, intellectually as well as just our own technology, that we don't. Sorry for my long-winded diatribe here, but I think in all this stuff, very often, when we're arguing over, oh, does natural selection, is it able to see what the errors are? And I'm like, we don't even know if it's an error or not, right? Because we're now learning that, oh, this thing that we thought has absolutely no function, oh, wait, turns out that's a big deal. Like, for example, the exists protein structure, it literally determines male and female. It literally like turns on and off the Y chromosome. Okay, it's like this whole protein complex, right? So we have a protein complex, a big one, that delineates like male or female. And so to me, when you kind of think about that sort of stuff, it's like, so for years we're being told this has absolutely no function, it's a bunch of junk. Oh wait, turns out it decides if you're a man or a woman. And then we're going to go argue over like, oh, this is fitness. Like, dude, you don't even know what fit is. Anyway, enough of my long-winded diatribe. It's something that's been running. That kind of stuff's been running through my head lately of some of these different things, listening to the same standard talking points from the Evo atheists, and I'm like, we don't even know what the heck's going on. Oh, and the last thing, I'll shut up, let you guys get back to it. Is I had a great conversation with Sal earlier today, and we were kind of going over some of the like protein sequence subdomains. And like the literal, we can now like map out all of most of the like subsets inside of the actual sequence itself. So it's like, oh, here's the overall sequence of the amino acids, and then oh wait, this 7 is, this helix. This is this strand. Oh, this is a turn here. Oh, this causes a curve. This is a hook. Oh, wait, we got a hinge here. And then we've got idiots running around being like, oh, well, you can have some errors and random mutations and it's no big deal. And it's like, bro, you have, like, we're mapping out like this 18 amino acids in a row. Oh, wait, this is the destination address after it is synthesized. And then it's taken to the endoplasmic reticulum. It's mapped out. Then they've cut off that tag after they put it into the shipping membrane, slapped on a new one, and sent it off to the destination point. And you guys are saying because that part gets cut off, that it has no significance and whether or not there's mutations in it? Like, you don't even know. We haven't even known what to look for.
Right.
Until very recently. But anyway, hope that all made sense. And I shut up now. Appreciate you guys.
So what you're saying is there's no possible way without male and female from the beginning that earlier you were talking about could even happen.
Basically.
Yeah.
Yeah.
Male and female same time.
Yeah, I mean, I was read a book that was published in 2020 on on epigenetics specifically, and this was there's probably like 2 chapters that they spent on that topic, and it was kind of like so we know that the short version is basically, we know this is what's going on, no idea how, and this basically explains why we can't. Like, they've done like hardcore experiments apparently for years trying to figure out a way to, because I mean, think about it, theoretically, it's if it's nothing but, a DNA, a chemical reaction, strands and all this jazz, right? Then theoretically, you should be able to take half of, male strand A and half of male strand B, try and bind them together in recombination. And theoretically, right, you should have the same outcome. Same thing with female.
Right.
And they're finding out that is a categorically false assumption. And it's just not being done. And the other thing too, that in terms of like the other epigenetic stuff we were talking about, you also have to remember that those modifications are being done to germline cells. Right. So it's like we have all these external variables that are going on. And then there's signals sent. And they've actually been tracking some of this stuff. It appears to be like signals directly from the brain, by the way. In some of the instances, are actually directing the modifications in the germline cells.
So when you were to Erica about this, she said, I don't know anything about genetics and biology. Are you telling us that she has faith in evolution then?
I would say it's a fair assumption. Yeah, I mean, because you got to think about like what are they finding? It's like some of the mice experiments, right? Like the ones that like get licked by their mother are much more like they're gonna lick their pups, right? Yeah, but then it dramatically lowers their stress levels and like all sorts of different potential benefits from the mother licking the pup. Then gets passed down. But then again, like I said, in kind of like more modern experiments, they're being able to actually track some of the things that are happening, and they're like they're watching like signals being sent and things being modified in the germline and new tags being done. Which to me, when you think about that, it continues to exacerbate the unavoidable reality that the vast majority of quote unquote observed like evolution is the result of real time or quasi or inside of the lifetime of the organism in question.
Right.
And it's like, so the mod happened then. That is kind of that's why, and Matt, I'd love to get your feedback on this, because I get so tired of hearing like, oh, well, the mutation didn't happen in the one organism, it happened in the population. I'm like, well, to get into the population, it had to have happened in a freaking individual organism at some point, right? Am I wrong on that?
Well, it has to be in the germline to spread throughout it. If it doesn't get there, I don't know how it's going to arise unless they actually believe the same mutation is going to cause the same problem in all the organisms at the same time.
Right. And to me, here's the other thing and Matt, I'd love your feedback, or SFT, I'd love your feedback on both what I'm about to say, because this has really been pissing me off lately. there's all of this talking point of mutations are caused by random copying error. But then we talk about environmental pressures, but then they want to claim that the environmental factors aren't what caused or were the primary cause of the replication error. But then in the experiments that are done, they're like irradiating an organism during its freaking replication and then be like, oh, there's a mutation. And I'm like, yeah, that's called an environmental factor.
Yeah.
But then if you actually go and like Google it, like read this stuff, oh, most mutations are caused by random copying error.
Right.
And it's like, wait, well, hold on. We have all these proofreading and error correcting components in play. And they are able to correct the vast majority of the replication errors. And the ones that are missing, it's like, why are they missing it? Is it a lack of nutrients, like a lack of resources? Is it additional external variables, i.e. radiation, for example, that is preventing the 100% correction rate? And do you follow where I'm going with this? Because I hear this, it's kind of like the whole population thing. I'm like, well, which one is it? They love to rely on, the environmental pressures and all these kind of things, and geographic separation, and it's like high level talking points, but then want to avoid on the molecular level what's the actual root cause. Does that all make sense?
Oh, big time. Big time. Yeah. That's the major problem with the evolutionists, is they don't want to focus on, as you were saying, what's going on down on a molecular level. We should be looking to the genotype as compared to the phenotype. Let's see what's really going on. And going back to what you were saying a while ago with the epigenetics, and what we see on a genomic level is essentially science fiction. We understand so little of the DNA language. We're learning more every single day when it comes to DNA function, right? Multifunctional and multi-purpose genes. Like you were saying, in the non-coding regions, when it comes to sex differentiation, right? That occurs in the non-coding DNA that the evolutionists assumed was junk at one point. Differential gene expression, when it comes to even your similar sequencings between humans and chimps, have vastly different functional purposes in terms of gene expression and epigenetic functionality. But going back to genetic entropy, everything you said, everything we know about the functionality of the genome, including epigenetics and epigenetic tags, means that genetic entropy is more of a problem than we really ever thought, because that means we are degenerating even faster, because that would indicate the majority of mutations, the majority of these typographical errors will then be deleterious, because they are hitting these functional regions of the genome, which often leads to a ripple effect, right? A typographical error here can affect a genetic sequence over here, because it's four-dimensional. So, I mean, overall, you said a lot there, Brother John, all good points, but I think the bigger picture is we understand so little of the genetic language, the DNA language. And in terms of testable predictions, we as creationists, intelligent design advocates, we are not scared to make predictions on genetic functionality, predictions on these long non-coding RNA, pseudogenes, ERVs, the non-coding DNA in general, because we expect design, we expect function. But the evolutionists, especially when it comes to this problem of genetic entropy, they need the majority of the genome to be junk, neutral junk. So those mutations that accumulate are absorbed by the area, making them 100% neutral. Well, now what we know, based on everything we've been talking about, based on all your great points, John, no, it would be difficult to demonstrate for certain that any mutation is just strictly neutral, no effects at all on genotype, So we just understand so little of the DNA language. It's mind-boggling. Go ahead, brother. Go ahead.
I would say SFP, I just sent you I'm on my phone. I just sent you an email. Will you show that image I sent you? So I think it kind of
yeah, let me go.
it encapsulates kind of what I was talking about. So as SFC is pulling this up, so this is a it's the sequence of a protein, but then you'll see it's all color coded, and on the left is kind of the key, and it shows like what's what. Pay attention. Not only there's like delineated regions, there's also this particular protein. Half of it is outside of the cell. Then there's a whole little section for like where it's passing through the cell membrane, and then there's a whole other section that's for the intracellular component or part of this protein. So we have a fully delineated protein sequence that results in a tool, half of which is outside of the protein, the other half's on the inside, and in the middle of it is like the little portion of it that like fits through the portal. Like
it doesn't even function without the other components.
Right. And I'm like, well, how do you get to this point? I mean, seriously, from like a rational analysis perspective, how do you conclude this? Like this sequence that results in this three-dimensional object. And the point I was making earlier about like this 13 is this function, and this 4 over here is a turn, and like this is what I'm talking about. And like, oh wait, turns out that these like individual amino acids, turns out like these different ones like interdispersed throughout the overall sequence, these are like hyper important, and what are they finding out in some of these, like, oh well, it turns out like all of these other ones, we could knock out like this, 10 here, 15 there. If we knock out this one, it's over. Like the whole thing collapses. It's done. It's toast. If you mess up this one. And you're like, okay, so you guys are arguing that somehow, sequence gets duplicated, and then somehow it's going to magically turn into one that's like, 7,000 amino acids long. And then in no way, shape, form, or fashion, any of the ones that we know are like, if this one has an error, game over. Somehow none of those got damaged or got mutated or messed up or misassigned, or there's a frame shift, whatever it might be. Like that, none of those had any problems, but somehow we managed to add 7,000 more. Like, come on, man! Like, I don't know what universe these guys are living in.
I know. Imagine even a single point mutation mess that region up.
Stan, did you get that image?
I've got the. Yeah, I'm gonna upload it as the overlay. So just 1 second. Computer's going a little slow right now, but I'm about to get it.
So did you?
There we go.
You doing Wi-Fi or did you plug it in internet?
Just once. It's probably lagging right now because I'm looking through my. I just saved this picture, but where the heck is it? Oh, here it is.
There, so look at that.
Okay,
like, get your
we're good.
Your C terminal at the bottom. You got all this stuff. Your start terminals, the repeats portion that's like part of the extracellular component. It's like the tumor or something. I forget what the TNFR stands for. Then you got your transmembrane. You see that little yellow section? Like that's the transmembrane component. That's the yellow part, and then the juxtamembrane. That's like a on the other side of the layer. Then you get the kinase, and then you got the C terminal. I mean, think about that. And then all the stuff that is the white lines underneath, like that delineates like hyper important individual or a couple of individual amino acids that are just like KO points. I mean, to me, when you look at that,
Yeah,
nothing about that even remotely looks random in any shape, form, or fashion. And then when you think about it from a engineering or coding perspective, it's like we have it's like a packet of data, number one, but it's got like, oh, here's the header, here's the start, here's the stop, here's the core function here, a little subset function there. Oh wait, let's packet it over here. Oh, then we got this little like end component over here, and then here's like some significant includes or calls to a database to make sure we've got the overall all the components that we need hooked together properly. I mean, and this is the protein, right? Then you have to think about it from then you got to think about it from the mRNA perspective, right? And the codons. And then you have to assume, I have to look at the sequence of this one, but you have to assume that there's introns and exons, or sorry, introns and the exons, right? So then you have that's got to be accounted for in order to get to this. And anyway, sorry for my long-winded diatribes, guys. This kind of stuff has been blowing my mind this past couple of days when I've been diving into it. And
oh, that's perfect, bro. That's perfect. That's why so many good points. That's why, where do I even start? That's why these evolutionists looking to these replication errors, these mutations to drive the evolutionary process, or in terms of genetic entropy, look to these beneficial mutations to counterbalance the damage. Based on all of this, it's like what they're looking to. It's like trying to fix a computer with a hammer. It's not going to happen. And not only that, is they have to explain the origin of this multi-dimensional, nested, multifunctional genomic information system, essentially, right? And that's why I always point out how little we understand, how little we know about the DNA language, where you said it earlier, John, and correct me if I misrepresent what you're saying, but they'll look to like a mutation, right? And assume that it is a mutation, but based on what we now know about the DNA language, essentially, a lot of this is not mutation related, right? It's due to built-in mechanisms for these types of adaptive events and episodes. And that's why this whole notion that the evolutionists look to the origin of all genetic diversity and genetic functionality as being the result of mutations over time and genetic errors over time, essentially errors in the nucleotide sequence. It's such an erroneous assumption based on what we now know about the function of the genome, that it's far more plausible to start from the design diversity position, or what you call, John, right, front-loaded capacity. That's what we see. Where they see mutations, we see design. And that's why I believe. Yeah, go ahead.
Right, you still have I think you still have that image overlaid for the rest of us. Or maybe my screen's frozen. I agree with you. The point, I guess, here's the way I look at it. So I agree with pretty much everything you just said. I don't think the mutations don't happen. I just think the mutations are, as we talk about all the time, like they're almost always negative.
Right.
Almost
yeah.
99 and it's agreed upon, right? That 99.999% of the time they're not beneficial. So if that's the case, then is it more plausible that the ones that are quote unquote beneficial are either actually the point you just made of pre-existing capacity that just requires a slight modification in order to be expressed, or the result of controlled modification, whether it be through some sort of epigenetic variation or other information processing capacity that's available inside of a cell. To me, that seems much more plausible, because when we're looking at adaptations, either you have to, and the point we were talking, Matt, you and I were talking about a few minutes ago in relation to this stuff appears to be happening like in real time.
Yeah.
And taking advantage of these functions, or it's happening like in one generation, right? The gets activated in parent and then passed on, usually through epigenetics to kid. I know one of the things you talk about regularly is the I think it was the people like in the Himalayas and stuff that have the crazy ability to operate at a high level in the low oxygen environment and all that.
Yeah.
But what happens when you take them out of that environment?
It turns off, goes away, like they never got it.
But if they go back,
turns right back on again.
Right. So it's like, did they adapt
right
from the evolutionary model, or did they adapt through the quasi AI model of? And because when I talk about that in terms of quasi AI, I mean like basically what we're trying to do is account for as many possible hypotheticals, right? And have those instances pre-programmed. Like that's what we're doing in our systems right now. We're trying to be able to have it like, write code on the fly and stuff, but we suck at that for the most part. Trying, but that used to be like the main focus. Like, hey, we're going to figure out a way to make our AI systems and do machine learning and like write new code function on the fly. It's going to be awesome. Yeah, well, that didn't work out too well. So now they've kind of shifted back. I mean, people are still being attempted, but now they kind of shifted back to like, okay, let's account for as many possibilities as possible, and then allow for the potential, like, machine learning execution of multiples of these that might have similarities in order to solve a previously unencountered problem by doing a combination of pre-existing functions, right? That might not necessarily solve the problem individually, but both of them might, right? That's one of the things that's being tried in like modern AI systems and such. But what the hell do we see in organisms?
Pre-existing function, exactly. Like
that plus we see two different, like we might see. I'm using dumbed down examples, like, oh well, protein A doesn't solve the entire problem, but if protein A and protein B both go after it, then it's solved. That's kind of what I'm talking about. Like you have a pre-existing. Both of them had pre-existing capacity and function, but then to the point that I know SNT talks about this a lot on you too, Matt. Like the whole moonlighting of proteins. Yeah. Well, is it a new function, or is it kind of like a sub function that wasn't normally turned on for that a sub capacity that was already pre-programmed into that, not as the primary function of that protein sequence, but as a if then.
Right.
And to me, when you really start to dive more and more into the like a little while ago, right before I came on here, I was reading a paper on the digital and the analog information in DNA, and how they've been realizing that there is a analog. There's the digital portion of the sequence, and then there's an analog portion of the supercoil that is being expressed and influences transcription as an analog while the digital is being transcribed.
Incredible.
So when you think about that, right, basically the conclusion they were reaching is that the chromatin is also operating, when it's a machine, but it's operating as an additional information component of the overall gene expression functions
at the same time.
Yes. No, not just at the same time, but like needed.
Oh, got it.
Like it's a component of the transcription, right? It's not the digital portion, but it's an analog portion that's also working basically in harmony or in unison with the digital transcription.
Incredible.
And this is relatively, like they've had ideas of this, but now it's really starting to be like delineated. And so when you think about that, another that's one aspect of another thing that's starting to gain some traction, which is the fact that there's multiplexing going on in genetic code or DNA in general, right? So you got the supercoil portion of the analog, you got the digital. And then they're also finding out that the physical shape of the physical three-dimensional shape is also having an additional information component in relation to overall protein synthesis. So you've got the actual sequence and is it the right? Basically for so long we've like, oh, it's the amino acid. That's it. No, no, not even close. Anyway, this one paper I was reading on that a couple days ago in relation to multiplexing, like they're basically having their minds blown because it's been very, very recently that humans have really been even truly starting to. We've known about the concept of multiplexing, like, hey, basically I'm going to send multiple signals down the same channel, right? And we've been doing that for a while, but that whole arena of like telecommunication stuff is really starting to explode. Basically, as we're running into issues with channel capacity for data transfer, we'd have to get more and more creative on how much information we cram down the same channel without conflict. And take it, maximize the capacity, right? On different frequency levels, all that kind of stuff, right? Well, then as our human knowledge of that concept has grown, people kind of like, hey, I wonder if the same thing's happening in DNA. Oh wait, turns out it is. And now they've been able to apply a lot of the principles that have been being developed in our normal communication systems. And now they're like, wow, it's like maximally optimized multiplexing capacity in DNA. Holy crap. And then now we're finding out, oh wait, that seems to have this is like another layer of information transfer that we didn't even know to look for, number one. Number two, we now realize it's there. And number three, now we're having to figure out like, oh, how much is this influencing the end outcome of the polypeptide? And this is why for me, when we listen to the Evos go on their tangents about like gene duplication, and modification resulting in new protein function and stuff. I'm like, dude, you have to account for three-dimensional structure, supercoiling, and sequence information somehow all magically happening to come into existence in an undirected process through random freaking mutation. Like,
right.
That's what you're arguing. Like, how is this plausible?
And these are just the layers we've discovered recently. How many more might be underlying layers beneath those?
Well, absolutely. And and by the way, the whole tangent I just went on, it doesn't even account for like alternative splicing.
Right.
And like all that stuff. And then the fact that like, oh wait, we're finding out that like introns actually have relevance in relation to the mRNA. And then untranslated regions also have relevance in terms of like the end fold of proteins and like influence chaperone action and all that kind of stuff. Like, we don't have a clue. Like, we do not have a clue about, in my opinion, anyway, like what's going on. I'm not suggesting we don't have, rapidly growing knowledge and all that kind of stuff. I'm not saying we're just complete idiots. But I mean, to me, when you really start to contemplate this stuff I'm talking about from the macro view, not just like, oh, this one little sub function, that's cool, no, that's really awesome. I'm talking from like the macro view, like this is what we're talking about in relation to life. And we're talking about just one of the mind-blowing requirements for like humans for us to exist, right? Like there's a whole neural code base. I was reading about this recently. Like, so I go on my tangents about genetic code all the time. There's a neural code that they're just now starting to like delineate of like how data is being stored in your neurons. And it's not in DNA sequences. It's like a whole new code base. Well, I'd say it's new. It's been in existence for a while, but I mean, it's new to us.
Yeah.
And I'm like, we have a neural data storing code base. What? I guess it's a necessary. I mean, it makes sense, right? That it would be there, but Anyway, I've been running my mouth for a while. I hope all that makes sense. It's been I've been having my mind blown quite a bit lately.
John, you lost us at hello, brother. Wait a minute, John. I think I get what you're saying, but can you start right from the beginning again and repeat everything you just said, because I wasn't really paying attention. I'm just kidding. No, seriously though. Okay, John, go ahead. If you got to come back for that, brother, go ahead.
No, I'm gonna resist. Go ahead.
Yeah, his channel. Yeah.
No, but seriously, so many points. Like, I don't even know where to start, because it makes me cringe. Okay, based on everything you're saying, John, based on everything we're covering, it makes me cringe when the critics, for example, when you were debating Gutsick Gibbon, okay, brother, when you were debating Erica, you were pointing out the functionality of the genome and how we are learning more and more every single day, okay? And her argument was, well, the more we learn still fits in that very small portion of the genome that is functional, between 10 and 20%. And the more we learn decreases. So therefore, there can't be any more than, 10 to 20% of the genome being functional. And to me, that's just mind-boggling, that how little we understand of the DNA language, as you're saying, John. Okay. How little we understand about embryonic development and the developmental process. Okay. How you've got, the coding DNA and you've got the non-coding DNA, and they're coordinating when, where, and literally how structures are sequentially being put into place to produce the fully functional organism, right? From embryo to fully functional organism. I mean, the non-coding DNA, John, literally functions much like a computer. But all of this came about by chance for no reason at all. Okay. And that's why I'll often point to the forward-thinking mechanisms in the genome, and I'll ask the critics, how can natural selection, which sees the short term, build that which requires long-term and forward thinking? How does evolutionary mechanisms build essentially a spare tire? But I'm just shocked that the critics can. Yeah, what are your thoughts on that, John? When you debated Erica and she was saying that, no more the genome is 10 to 20% functional, and everything you're talking about in terms of genetic functionality, it's still all within that 10 to 20%. Therefore, most of the genome is junk. I mean, how can an evolutionist come to that conclusion, John?
Honestly, I don't know how evolutionists come to any of their conclusions. They make no sense. They're counterintuitive and require willful suspension of rationality, especially given what we now know. I mean, that's where it comes down to for me. I mean, like, I was watching a lecture from Dr. Howard Chang from Harvard or Stanford. It's from like 2 years ago, and he was on a full exposition about a variety of topics. But one of the things he was covering was the significance of the gene clusters, and then how there's all these there's all this non-coding portions, and then there's another cluster, right? And then nothing, quote unquote nothing, and then another cluster. Well, from the purely linear perspective, it seems to make no sense, right? But from the three-dimensional perspective, it does. Because when you account for all the folding, oftentimes it's putting these clusters on different chromosomes, right? Putting them in the appropriate physical three-dimensional locations for transcription factories and other regulatory components that otherwise would be way far away, right? So from a purely linear perspective, they are super far away. It's a million bases, between these other ones. But from the three-dimensional perspective, they're now literally next to each other, right? from a coiling
Right.
perspective. And and then when you factor in like, you know, all the histone modifications and that nature, yeah, there's this blank area, quote unquote, but it actually enables the appropriate coiling to be allow for easy access, and then recompression of it. So, but then simultaneously, if you have you've uncoiled this one region, now this supposedly junk portion that, you know, repeats or whatever, are able to bend in an appropriate way to now interact with a transcription region. And now expression can be transcription and expression can be executed. And then what are they finding now? That damage mutations, like even minor shortening or lengthening of some of these supposedly non-functional regions, completely jacks up a whole segment of DNA of genes. Like, we know this. This is known beyond doubt now, that there are quite a few actually genetic diseases that are literally the result of, you know, what's his face, Downard. What's he love to go on a tangent about short tandem repeats? Like,
ALUs.
yeah, proof of evolution, right? What do we now know? There's all sorts of genetic diseases that are caused by, like, oh, well, normally you have between 30 and, you know, as long as you're within 30 to 50 of these repeats, you know, before here, you're good. If you have less than that or more than that, you're toast and you have XYZ disease. Why? Oh, wait, because it jacked up the fold, the three-dimensional fold, and now this gene can't be expressed. So, does it have function or not have function? In this context, the function is literally the three-dimensional movement component in order to enable the execution of that function, or, you know, of the coding function. Well, I mean, this is like, I mean, I don't even know a good analogy off the top of my head of like how to explain that, because our tech isn't that good, right? Like, we don't have that capacity to do this, and yet we've got people like Erica going on tangents about how, like, oh, it's not coding, quote unquote. Therefore, the rest of it's junk. It's like, okay, so if you go on the other piece, it doesn't do anything, and actually has a negative outcome. So I'm really not following your logic here, if there is any. Does that make sense?
Well, this is why what you were saying earlier about how they looked at gene duplication followed by neo-functionalization, right? You'll hear them appeal to this in terms of how new functions can arise and a net gain in information, for example. And they'll look to these gene families, right, in organisms, and typically based on phylogenetic assumptions, phylogeny-based assumptions, they'll assume that these novel gene families were the result of gene duplication followed by neo-functionalization. And they think that these types of mutations have no real deleterious effect, okay? But what you're pointing out here in the fact that the DNA code is not just a linear string of letters. It's what? It's multidimensional. There exists overlapping codes. It's self-regulating, okay? So where you have a mutation, let's say a gene duplication, point mutation, frame shift, whatever, you name it, okay? In a genome that's nested, self-regulating, and multidimensional, you are going to have a ripple effect of problems. So this assumption that this gene duplication followed by neo-functionalization can lead to evolutionary advancement, novel structures, new functions, it's based on a faulty assumption. You know, it's mind-boggling to me as to the story and the imagination of these guys. Go ahead.
Well, and something else in line with what you're talking about there in terms of non-coding. Like, non-coding is relative. So, I mean, you've got circuits, right? You've got on and off switches. Go and Google, somebody, just for entertainment at some point, Google source code of the original Pong. And what you'll find out is there, for the original version of Pong, way back in the day, there was no source code. You know why? It was all nested circuits. That game was enabled purely through the formation of on and off circuits. There's a crazy diagram you can see of it, which ironically looks extraordinarily similar to like some of the the portions of the genome, which is a sidebar. But the point I'm making is the on and off switches. I mean, how do you think that binary is expressed, you idiots? Like, we have circuits. Hello, we've got on, off, and or nor, right? And then we're trying to get creative with some newer ones, right? But what we've been finding out is that because we got a quaternary versus a binary model, the genome actually has the capacity for more variable on-off or more variable circuit switches than what ours do. Well, some of our more modern ones we're getting closer, but the been around for billions of years functionality, right? The on-off switches are more advanced than what we've got in our computers right now. They have the ability. Okay, so think about this in the context of, upregulation and downregulation. What the hell does that mean? When you like expand on that for a second, like up and down regulation. What's that? What's that mean in general?
Oh, just I was going through my emails. Okay, are you asking us what does up and down regulation mean in the sense of
in the genome? Like when we hear like up regulation, down regulation, like what are we talking about of a gene? Okay, so based on so if the gene's being upregulated, right? There's a promoter region, then an operon, all that stuff that's accounting for, like at the lac operon, right? It's the lactose is fit into that switch, right? And when it's there, it's active, I guess. And when it's gone, it turns off, right? That's an example. But you can, and then based on the external variables, then more or less is going to be being produced. It's not necessarily going to just be on or off, right? It can be on and being a bunch of it being expressed, or it can be off and then we'll turn on and have a little bit, right? We can have variation of the amount that's actually being expressed. Right. How does that happen? It's happening because we have variable control logic gates for the regulation of that particular gene or genes. And then when you look at the cascade effects of there's a switch that controls this gene up here. But then if that gene is activated, then it's producing proteins that go and turn on or go express or act as the recruiters and such to express these other genes that are the result of the switch for this other one who might be way away, not even remote, not even close proximity. It can be you can be a million, it can be a freaking different chromosome for all that matter. And now it is resulting in the expression of all these other genes. And that's to me, that's some of the things that really start to get into the level of how do you explain this without foreknowledge? I don't know how it's remotely plausible to conclude that gene A helps to, or is a primary factor in the expression of genes B, C, and D. However, it's not in remote physical proximity, and it has an on-off switch that turns on proteins that then go and activate these other genes that are not in physical proximity. I don't understand how just the whole it's chemical reactions and undirected mutations and such. I don't know how that's remotely plausible.
Right.
To come to that conclusion, because there is no cascade effect. I'm talking about in the pure, just chemical reactions. It's not there. So how do you explain that?
Right. Well, from the evolutionary perspective, where they're looking to natural selection and mutations, essentially they can't explain it without invoking a mind for evolutionary mechanisms, because you're right, it does require forward thinking. Just like you're talking about gene regulation, just gene regulation in general, where you've got control, right? Just the fact that there's control of which genes are being expressed and how much is being expressed. Different variations, I guess, like you're saying, in the amount of the product that's being expressed requires a mind, requires control, requires forward thinking. I've asked that question numerous times during debates as to how can you account for that which requires forward thinking without a forward thinker. Oh, these neutral mutations build up over time and they're subject to genetic drift, and eventually they're called upon in order to evolve these types of functions that only look like forward thinking, but they're not actually the result of a forward thinker. They're just a result of, mutations building up over time. They're kept in this hidden reservoir that they want to say can be called upon in the future for evolutionary advancement. But, they can get around it any way they want. But essentially, they're giving selection a mind, and selection sees the short term. Selection doesn't see the long term. How does selection know that these neutral mutations are one day going to be called upon to evolve these types of controlling mechanisms, these types of epigenetic mechanisms? I don't know. That's what comes to mind based on what you're saying, brother. But it's mine. Yeah, go ahead.
I think one of the things that we really have to think about, I know I at least intend to kind of start focusing on this a little bit more, is the mechanics of the things that they're claiming came into existence through undirected process, right? Like, we are talking about three-dimensional molecular tools and nanotech machines. Like, this is an unavoidable reality of where we're at now. Like, we know this. We have the technology in order to observe these things in real time. Not all of them, but I mean, we're getting close to the real time all the time. But like, we know what they're doing. It's a pair of scissors. It's a screwdriver. It's a drill. I mean, it's like, these things are amazing. And I mean, ladies and gentlemen, we have transportation trucks that move at the equivalent of 400 miles an hour on roads that form in front of them in real time. Like, this is not figurative. This is not hypothetical. This is not just imagination. Like, this is a recognized premise. They're traveling the equivalent of 400 miles an hour, zipping around with a, trailer they're pulling that's loaded up with stuff that has logistic tags on it, and run through like a warehouse and then get shipped off to another place after it gets loaded onto another truck. Like, I'm not making this up. I mean, like, this is actually what's happening. And then you think about stuff like for transcription. I mean, you've got 85 in your body right now. There's no less than 85 different little nano robots that are required in order to perform gene duplication. 85. One of which has a RPM rotation of 20,000 per second. Like, we're talking things that are better than jet engines. Like, I mean, to me, when you think about that kind of stuff, it's like, what are we arguing over right now?
Right.
Okay, my position, which one of us believes in the Sky Daddy? I mean, let's be blunt. We've got nanotech robots that are running around doing crazy stuff that is only in like sci-fi, like human sci-fi right now. And it's real world stuff. And on one side, I'm told that this must have come into existence through undirected process through deep time. And on the other one, it's like, so we've got computer software, we've got hardware, we've got computation, we've got information processing, we've got nanotech, we've got ridiculously badass robots, and it's all working in unison. Oh, and we've got all of these engineering components that are necessary for us to exist that are literally optimized to the maximum degree possible based on the laws of physics and chemistry. So, like, there's parts of our body, in spite of all the ad nauseum talking points from the evolutionists out where it's just badly designed. It's like, okay, well, explain the fact that it literally cannot get better. Like,
Right.
and Sal's talked about this, like the calcium ion channel, for example. In terms of physics, it is impossible for it to be any better. Like, it cannot be. There is nothing, like, it can't get any smaller. It can't get any more efficient. Like, that's where it's at. Yeah,
Right.
you think about he's talked, I know Sal's also mentioned like the sharks and stuff. Like, their sensors cannot be improved in terms of physics. There is nothing more sensitive that's possible in relation to physics in terms of what they are able to sense. Like, it cannot be any greater. And that's a recognized premise. Like, so you're telling me we're horribly designed, except we have things that are to the max that cannot be better than, like, so we're just evolved all the way? Come on now. Let's get real. On the flip side, you're going to tell me that, everything else is, examples of, just scrambled crap. I mean, and the far from my tangent here, but like, what's the having a brain freeze? What's the nerve they always love to go on their tangents about, that the giraffe? We all have it, but the giraffes.
The vagus nerve, I think.
Yeah. Okay. So for anybody who's listening who has never actually looked this up, do not listen to that moronic, ignorant, uninformed, outdated atheist talking point. Okay. So twofold. There's multiple types of nerves. Number one, it is a some nerves only have one type of function. Others have multiple. It's a multifunctional nerve. Also, the whole talking point about why is it going this long way around and come back up to your larynx? Why doesn't it come directly from your brain? Well, idiots, if you go and actually look at it, there is a nerve that goes directly from your brain to your larynx and is the primary for speech. That the other nerve is going down and connects to no less than 4 other organs before it comes back up and then reconnects. It appears that twofold. Number one, and they know this beyond all doubt, by the way. For example, if that nerve gets damaged, you're likely to have heart failure. Just as an example. It's a multifunctional nerve. But it also can serve as a backup for your speech. You have major damage to the one that's coming directly from your brain to your vocal cords, but the other one's not damaged. Guess what? You can still talk. So it's like it serves both as its like they've misinterpreted what the primary function was. Primary function is to be going and connecting with all these other major organs and having input-output that's happening there, and then also act as a backup for your communication method. It's a 2-for-1, or in this context, like 5-to-1 layer of functionality. And yet, what do we hear? Ad nauseum. I mean, I think I've heard it from at least, well, I'd say no less than 5 Evos in the last 6 months on debates on your channel. I mean, it's the same talking point. It's like, how many times do I have to tell you this? Like, this is well documented. And yet we hear the exact same ongoing talking points from these guys, and it doesn't matter how many times we tell them, they repeat the same thing over and over again. When we ask them if they gone and researched what we told them, 2 months later or the next live stream, and they're spewing the same talking point, we say, hey, have you gone and researched what we told you about? No, I didn't.
No, they didn't. No.
So you didn't research it. You're claiming that you're disproving my position when you didn't actually go and check up on the point that I made, the debunks your talking point. And then very often they're going to claim that we don't actually go do research. Show me the literature. Well, every time I show you the literature, I'll tell you what to go look for. You don't go and do it, and then you claim I don't have a point. So to me, we're kind of reaching the level of, let's just inform people that have open minds, give them the direction. And I'm like, hey, go look here, go look there. Here's what we found. And then not even try and convince the Evos because they have no intention of being convinced. And by that, I mean, I'm talking about like the activists, not the just the average person who's been told this but hasn't really looked it up for themselves, you know?
Right. Like your militant atheist evolutionists that we see online, of course, that make it their job and their life mission to to reject or disprove in their eyes an intelligent designer. But going back to a couple things you said that are on my mind, one thing like these backup structures, right? It makes me think how many times the evolutionary community has had egg on their face, essentially, John, because you're pointing out an example, right? This specific nerve. You're pointing out an example of what the evolutionists, especially the apologists, look to as bad design. Why would God design this way? And 100 years ago, the evolutionists looked to, I believe it was over 140 vestigial organs, no function. Okay. Now they've revised their definition and have changed it to lost its original function, or maybe it's co-opted a new function, but that's revision. Okay. It wasn't like that 100 years ago. They really believed that there was over 140 organs that had no function. Now, with what you're pointing out, we're finding out that these structures and organs that they used to look to as non-functional, or they look to as bad design, have secondary functions, or it's like a 5-in-1, as you pointed to. And it reminds me of the backup code that we actually have in the genome. These redundant mechanisms that any computer coder, you know more about that than I do, would look to as good design. Right? Redundancy is good design. It's forward thinking, like these third position codons, right? This third position codon variation that they say is redundant. And why would a designer use this type of redundancy? And they can tolerate error because it's not going to have any effect on the actual protein. But now we're finding that mutations to these redundant elements can actually affect the slowing down or speeding up of these processes in the cell. They're actually involved in information flow. So yeah, redundancy is good, but the more you mutate these redundant elements, like in the third position codon variation, it kind of goes back to that paper that you and I both love on translational pausing. So it turns out even these redundant elements are not strictly neutral, because it's still having an effect on the processes in the cell when it comes to information flow, for example. So everything in the genome is functional, and everything in the genome, because you said you're looking at a perfect state. What did you bring up? The calcium ion channel that Sal points to, or even what's that enzyme that Brother Sal points to? Topoisomerase. So all you can do,
there's more than one. There's two of them. They do
right.
One cuts all the way through, one cuts part.
Right.
You have to have both of them, otherwise you have nothing.
So what this means is it can't get any better. It can only go downhill. Even in these redundant mechanisms and this backup code and backup structures, you damage the backup structure, you damage the backup code. You're only losing more of the initial front loading when it comes to the redundancy. It's like the treading on your tires of your car. Yeah, the treading's only going to be there for so long. It's there, and it withstands damage, but it's only going to be there it's only downhill. I mean, there's no gain. There's no getting it any better. The calcium ion channel you pointed to is not going to get any better. It's there. It's designed. It's perfect. It's only downhill. Go ahead, brother.
Well, and the point you were making about the wobble effect, I'm honestly starting to get massive levels of entertainment when Evo atheist idiots bring that up. Okay. Guys, go read up on this, because you are ignorant fools. We now know that the ribosome is being able to read in context, like the codons before and after. Remember, there's like 3 codons in the Actually, it's like 3 to 4. I think it's three in the 3 positions, right? Coming as the mRNA is coming into the ribosome. We've actually been discovering that the ribosome is being able to quote unquote read the codons in proximity, right? And which codons are next, I think it's up to 3 on either side, are being able to influence the correct or incorrect interpretation of a codon in the center, right? So it turns out that part of the reason that the wobble effect is even able to be done is that there's the capacity for being able to read things from a contextual perspective. Context, folks, context. Which, let's think about this. If you have a word and you have put an asterisk in for one of the letters, are you probably going to be able to figure out what the actual word is if you have the other letters? Yes, you are. If you have a brain, that is. This is what we're talking about, folks. Yes, we can have a wobble effect. It's like having a letter in a word with one letter's error. You're still able to know exactly what that word is based on the context of things surrounding it. That is what we now know. We know this is true. So don't act like the wobble effect is somehow an example of bad design. It's actually enabling faster execution and allowing for error to be executed on the fly while being corrected on the back end. Like, this is what is happening. We know this. Oh, and how do we know this? Wait, it's because it's exactly what we do in our systems. You uninformed morons that spew this talking point. We do this. We do this in our communication systems because we know this is going to happen, and we know that if you want to have speed, you have to account for that on the front end to be like, okay, I know this is going to happen. Therefore, I'm going to account for fixing it on the back end. And, oh, wait, that's exactly what's going on in the ribosome. And we have the functions and the different components in there that are necessary in order to execute that. On top of the fact that, this is a sidebar, man. Did you know that every single tRNA and amino acid actually has an additional enzyme that is specific to it as part of the reading process? But we forget about that, right? So you have the codon, you have the tRNA with the anticodon, you get the aminoacyl, right? And then you have an additional enzyme, a specific one for each type.
Right.
We've got all these things that are working in unison to do proofreading, to make sure it's the right stuff, like multiple fact checking. And they want to argue over like, oh, cool, I've got the capacity to be able to account for not even have every single component of this on the fly in order to maintain the fact, oh, wait, I'm transcribing things at thousands a second. Seriously, let's think about that for a second of what they're acting is like I think this is inferior. Dude, we're processing so much information that, why do you think that we are working on cellular computers? It's because you think about having all these ribosomes being able to process all that information, like individually, not as fast as a computer, collectively, real fast. The amount of capacity for information processing is mind blowing. For example, you got a beer, you get yeast, right? In a pint of beer, there is more information processing capacity than in a supercomputer. In just the yeast in one pint of beer, vastly superior to our best supercomputers in terms of like the actual amount of information it can process.
Unbelievable.
We don't think about this stuff, right? But then we listen to all these uninformed Evos run around claiming that it's all examples of bad design, and it's like, your uninformed status does not negate reality.
Well, that's why they're always shocked, and that's why they're never predicting these, like, for example, they're always shocked when we find how incredibly functional and essential, let's say, the endogenous retroviruses are, and these other classes of retrotransposons, pseudogenes, we now know are incredibly functional for healthy processes in the cell. The ALUs that James Downard loves, right? These highly functional genomic networks that we're finding across genomes. They're always shocked. It wasn't expected, wow, it's amazing what evolutionary processes can do. And they're not making, the biggest thing is they're not making any specific predictions on, like you were talking about earlier, on how mutations to certain portions of the non-coding regions of the genome can lead to disease, right? Well, as intelligent design advocates, there are specific predictions out, like, hey, you mutate this genetic sequence or this DNA position, it's going to result in this disease. We expect, because of the starting point of design, we expect treasure in the genome. Well, they expect junk. So they're just going to be surprised more and more, and it's just going to be more egg on their face when it comes to, like, for example, the junk DNA paradigm is like the new vestigial organ argument, right? Back 100 years ago, when they thought over 140 were just useless, now you got the Todds of the world that still use that argument, but most evolutionists, they'll shy away from that because it was premature thinking. And now the same thing is going on in the genome, where now they're assuming all of this junk, genomic fossils, evolutionary leftovers, ancient viral infections. No, it's all there for a purpose, there for a design, there for function. Before I go on for too long, brother Jamie, good to see you. Good to see you. Always a pleasure. What's going on, my man? What's going on?
It's all good. Just listening in.
That'll boy. I appreciate the Superman video you sent me. I loved it. Had me cracking up. You're good with those, John. Jamie's been taking.
Dude, I'm very aware. He
knows.
I've got like 10 of them in my inbox. I think Jamie's like become addicted to like Face Off, whatever that app's called. And,
yeah, it's no skill involved.
Yeah, he's been having a grand old time with that one. I'd say that much. It's and I've been, let's see, you sent me recently. I am now Arnold Schwarzenegger from Terminator.
That one's awesome.
Yeah, I'm Xerxes from 300. That's a pretty good one. I have to admit, that's a good one. There's a bunch of them. Yeah, I mean, it's Trump. It's a bunch. Yeah, it's good stuff. But yeah, all this stuff, man. It's like, what is the sheer desperation to avoid the obvious is really, it's a conclusion that I'm coming to, of the desperation to avoid the accountability that is the logical conclusion to the necessity of a designer, right? Like, okay, if the designer exists, therefore an entity higher than me and preceding me must exist. Therefore, I cannot refuse any accountability other than what I decide for myself. And at the end of the day, I feel like we're dealing with a bunch of rebellious children who are desperate to, refuse concession to parental authority, just for example. And I don't mean to put myself in as the parent. I mean, God obviously is the parent. It's like, I don't know, man. Like, at what point, like, are you guys gonna just, come back down to the real world? Like,
Right.
one of the times is myself as a teenager, and then in college, I had my sense of immortality and all that kind of stuff, and I was doing my thing, and then even in my early 20s, still had that feeling, and I started to grow up a little bit, and then off doing business, and I was growing up, and theoretically from like the worldly perspective, oh yeah, doing business and all this kind of stuff, but from a like true, a real level of maturity in terms of like my existence itself, I mean, it went until my late 20s. And, when I think back to all the stuff that happened to me, if I hadn't gone through what I went through, would I still be thinking about my own existence from that same arrogant and, almost juvenile perspective? And
Right.
I feel like that's kind of what we're dealing with a lot here.
It's almost lucky to find that, have that experience of facing death, because it makes you stop and realize what's at stake. Exactly.
It makes you question what's more important than where did we come from? What's our origins? These types of questions. Why are we here? And a lot of your rebellious children, like you're saying, instead of seeking answers for those questions, they'd rather rebel or reject or ignore or not even think about it. Most people just don't even think about it. They don't care. They're just busy with their life and living according to the world. But then your militant atheists, they want to fight against the reason behind our existence. And as John was saying, sometimes it takes those moments or situations or experiences that are those types of negative experiences, I guess, that I think God uses to bring us to him in that we will now kind of question, to start seeking answers to those questions. And one thing I wanted to say for the audience sake too, based on everything we were I know that we touched on a lot of information there, me and John, for a while, on genomic functionality and forward thinking mechanisms. And just in case some people are thinking, hey, this is creationist talking points, everything we're saying here, and as usual, I've got paper upon paper here just to show that, hey, listen, this isn't just things we're making up from a creationist perspective or just getting from creationist websites. I mean, these are from the secular journals themselves, like Pseudogenes. Even some of your theistic evolutionists like William Lane Craig, they're guilty of evolutionary assumptions, evolutionary arguments, and starting points, because he did a video recently where he was saying that, pseudogenes are the best evidence for evolution. So that's why I believe humans and chimpanzees share a common ancestry. That's why I'm a theistic evolutionist. And yet even somebody like him is not aware of the overturning of pseudogenes. I mean, all these just right here, 1, 2, 3, 4, 5 papers about the amazing functions in pseudogenes. For example, all your endogenous retroviruses, retroviral promoters in the human genome. John and I were talking about intronic ALUs, the ALUs that RJ Downer loves so much. They're being used to influence alternative splicing. Right here, we were talking about this for a while. Redundancy of the genetic code enables translational pausing. Okay, secular journals. We're not just making this stuff up. They're baffled. Protein moonlighting, these multifunctional proteins, where, what was it, 50 years ago, John? More than that, probably. The paradigm was one gene, one protein, one function. That entire paradigm has been completely overturned. What we see in terms of these proteins are like a multifunctional tool, like a Swiss Army knife, where you've got, all these different tools in one for different functions and purposes. That's what we're seeing with these proteins. Introns, orphan genes, I mean, don't even get us started there. I like your questioning with rational empiricists the other night, John, where their explanations for orphan genes, these taxonomically restricted genes, they all fail. It's all just a series of hypotheticals, maybe this, maybe that. But you would need at least 5 or 6 consecutive beneficial mutations to build something like an orphan gene. It's much easier to, like we were saying earlier, to break something down than build something up. So, I mean,
I think something else in line with that point you were making there. I kind of touched on this earlier too, but okay, we now know that these 5 amino acids, in relation to being after this strand or this helices, right, executes the turn mechanism in terms of the fold that then enables a new, and then if you now have another correct sequence inside of this protein, overall protein, right? To do the next helices, that's now gonna. Oh wait, there's another turn here, right? It's like, okay, cool, folks. We're now talking about not only do you have to have the modifications and how many modifications, you have to have the modifications in a very specific spot.
Right.
And I feel like a lot, even for the ones that are supposedly looking into this kind of stuff, like a rational empiricist or David Neff and some of the other ones, when you actually ask them, they haven't gone and looked at this for themselves. They like to stay at the high level theoretical talking points, high level evolutionary biology concepts, rather than actually look below the surface at the plausibility for themselves of, have you actually gone and looked at sequence A and sequence B and been like, that's a crap load of differences. And then been like, okay, I'm talking the overall sequence, right? And then be like, okay, how plausible is it that now I'm now looking at the subdomain of the overall sequence, right? Not only did this number of changes have to happen, they had to happen in these specific spots in order for this new functional protein to exist. That's kind of why that image I sent you, I was like pointing out like the things that were underlined in white.
Yeah.
It's like, those are recognized, like, oh, we've got to have those there. Otherwise, if those are jacked, we got nothing.
Yeah. Oh, that's being added to my slide.
So what's going on with that? Why do you think that these people, do you think they're just not looking at it that way and they don't even want to think, they don't want to go there? Or do you think it's like taboo to even think in those terms of like the source of this kind of thing? They just don't even want to go there. Like, what do you think the issue is there? I mean, we know it's a spiritual issue, but why is it taboo? Do you think they're scared about, well, if everybody accepts that and just the obvious, it's going to mean, theocracy and stuff?
Oh, that's a good question. I mean, I think some of it is I think some of it's fear, to be honest. And it might not even be conscious fear, just a underlying subconscious understanding that if they actually go and look at this, it's going to annihilate their worldview.
Yeah, there's probably different levels, right? People that are like ideological about it, and then there's like probably the majority that are sheepish about it. They don't want to be. They wouldn't want to speak up because it would bring shame or whatever.
Yeah, well, and let's just be blunt. They're not all, but a good percentage of, and this I mean I'm talking about the hardcore Evo activists. A good percentage of them rely on evolution to justify their atheist worldview.
Right.
And they love to deny it and claim it does. Oh, it doesn't actually have any bearing. Yeah, it does. It's obvious because they run around screaming about Sky Daddy whenever we talk about abiogenesis and the implausibility, and they make it like, you just believe in God and God of the gaps and all that kind of stuff. Oh, wait, but this has no bearing on whether or not I believe in God.
Well, remember this.
Okay, so that is not even remotely what you're acting like. Come on now.
And a lot of these militant atheists, like your Matt Dillahunties, Godless Engineers, Aaron Ross, what do they say? Even if they knew, if Jesus Christ presented himself to them, God came down and said, I exist. They have said that they would not worship him because they would not want to worship the God of the Bible. They don't like the God of the Bible.
Yeah.
So therefore, I don't want to judge everybody, but at least those militant atheists, I mean, they are starting from the assumption that there is no God. They're starting from a point where they don't even want to worship the Christian God, even if he showed up in front of them and said, hey, here I am. So all of this evidence that we're talking about, yeah, they'll fight it to the end, because that's their basic presupposition. There is no God. None of this is therefore due to design. None of this is the result of a designer. You can go as far as Richard Dawkins, who will say, yeah, the DNA code kind of looks like it's designed. So therefore, maybe it was designed by aliens, some type of intelligence elsewhere in the universe that came about through evolutionary mechanisms. He said that, which is kind of silly because
the chicken and egg problem.
Yeah, so that it just pushes the problem back further, because then where did that intelligence come from? You know, according to Richard Dawkins, some other evolutionary process brought that intelligence about. It's just like this never-ending kind of.
Hey, so quick sidebar, guys. So you know how, well, most of y'all know this, but occasionally I will, for purely entertainment purposes, because I can only take them in small doses, but I get a chuckle. It's almost guaranteed. I will jump over to Speed of Pudding's channel, and tonight was no different. Right? there were actually, I went back twice because there was so much entertainment value. But this guy, I think what I'm about to say just kind of encapsulates the point that Stanley was just making there, like just the sheer desperation, right? These idiots, and I say this, and I don't mean just like insult. I mean, like, I feel like I'm listening to full-scale idiots, okay? They were going on this whole tangent about how the Bible is disproven, because if we were actually made out of dirt, then we should be silicone-based, not carbon-based. And I'm like, wait a minute. I'm pretty sure that dirt's got a lot of carbon. So what do? I go and Google it. Sure enough, carbon is the primary component of dirt. So, yeah. So I'm just sitting there like, okay, so I've just listened. I mean, when I was listening to them, I was chuckling before I Googled it, you know, then I'm letting that start cracking up even more. But I'm like, so we've got 5 grown men on their computers searching for a desperate way to somehow disprove
beating a straw man to death.
Yeah,
right? In a literal, 2-second voice command on my phone while I was walking across my house, right? I'm like, is there carbon in dirt, right? And then sure enough, like, carbon is a primary component in organic soil, blah, 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 like all this stuff. And I'm like, so they're justifying their atheism and in their mind and their worldview negating Genesis because they think that dirt is made of silicone. Like,
I have a better one with you. It's Leo argument.
I don't even know where to go from this point. Like, I chuckled and then I couldn't take. They kept being stupid and I left. But go ahead, Will.
Have you ever heard legal argument against the existence of God? You won't be ready for that. Leo said that God doesn't exist because according to the Bible, God is outside of time, space, and matter. So therefore, God cannot be anywhere. There is no when and there is no is. Therefore, God doesn't exist. That's Leo argument against God. Isn't that stupid? I mean, those guys are ridiculous.
Those speed of sound streams are a comedy show.
Yes.
They're a train wreck and they're worthless. I mean, have you ever seen Speed of Sound attempt a rebuttal video to something we put out? They can't get through 30 seconds of a video. Like, yeah, go on.
Well, I think I'm about to say what you were saying, but I've been telling him, he's been whining about me not, addressing his points for 8 months now. And I have told him over and over again, create a short video, outline your argument, I will address it. And I think I'm guessing where you were going with this is they start these videos, maybe 30 seconds are actually on it, and then before you know it, they're off on some completely alternative ramble, and you're like, what are you guys even talking about? Like, okay, tonight, him and some other person, they were like whining about how they're they have effeminate tendencies, and then like Pudding was like talking about how one of his first memories was how he wanted to buy a cooking set, and his dad was mad, and because he wanted his son to want to buy a truck versus a cooking kit, like. And somehow this is like in relation to their atheism. I'm like, what am I listening to right now?
Yeah, it's bad. It's bad. I mean, yeah, they'll get maybe a minute. Yeah, maybe they'll get like a minute into like it doesn't matter. It could be a 5-minute video. It could be an hour video, 20-minute video. They'll get a minute into it and it's completely derailed into something completely off topic. And even the minute that they still address nothing, or it was a straw man or a misrepresentation. And speed of sound, I mean, none of them really have anything. Here's the thing. If the best they have is your Dan Stern Cardinals, then we should be very confident in our position because even the best that they have to offer don't really have much to offer. So yeah, those are comedy shows, Speed of Sound's channel. So,
well, and here's the thing. JP, I think Jamie was trying to say something, but
yeah.
Jamie, were you trying to interject?
No, go ahead.
That whole crew they crack me up, to be honest. I feel bad for them. I mean, it's like okay. I'll put it this way. I don't know if you noticed this, that this I'll get a couple days go by. I'll swing over. I'm like, oh man, I wonder what the Pudding channel's been up to. I'll swing over there to see what's going on. And it's like so literally every video is a review of like one of my after shows, your debates, somebody else's after show, like Otangelo, a stream on Otangelo's channel, and it's like, so basically all you guys have to work with is to whine about what we talked about. I'm just like, what is happening right now? I mean, maybe I should just feel egotistical and like allow my, my arrogance to expand, because apparently I have justified my own personal, like I'm one of 3 or 4 people that are like the primary focus of another channel to do reviews of. I mean, I guess I'm special. I don't know what to say, man. It's like, what is happening right now with these people? It's getting. I don't know, man. I kind of crack up about it, to be honest. But I tell you what, folks, if you haven't seen this, it's totally worthwhile. They literally titled one of their streams like something about Dumb and Dumber, and they've got Jim Carrey and Jeff Daniels as like the thumbnail from Dumb and Dumber. And I'm like,
send me their pictures and I'll make a good one.
Oh man,
you'd be good at that, Jamie.
Oh man, when you look at that, I remember when I saw that and I clicked in and I listened to them like talking like idiots, and I'm like, we have Dumb and Dumber. Like I was like, I don't know who's more dumb, like the guys doing the stream or the characters from the movie, because
oh yeah, you posted that, didn't you? I remember that.
Oh yeah, I posted in my community.
Like, this is literally what they said.
Who did this? Like, who would do this? I'm never ever. There's no way, shape, form, or fashion where I'd ever think it was like a remotely rational thing to do to project that I'm dumb and like I'm either Jim Carrey or Jeff Daniels from Dumb and Dumber. Like, I'm not going to insult myself in that fashion. I'm just not going to do it. I don't know how you would come to that conclusion. Other than
I was confused what that was saying.
Well, here's the thing. I think they were trying to project that onto. I forget who they were. I think they bounced around between who they were talking trash about, but there wasn't enough coherence between all of them to actually figure out what the topic was. So that's what I'm saying. Like,
that's the problem, though.
Like, you guys are the characters. You idiots are acting like the characters from the movie. It's like it's too good. You can't even make this stuff up, you know.
No, well, I was saying that I debunked a bunch of arguments last night from Dodgeball Dan and some of the Dodgeball crew, and I must have said that a few times. Like, you can't even make this stuff up, what we're dealing with here, and yeah, I don't know. I lost my train of thought there, but we've been going at this for a while. But that's what I was going to say. Like you were saying earlier, it's laughable, but also sad at the same time, right? Because you feel bad because you see how defeated they are, as in they've run out of talking points, which means they've circled back and they're repeating the same talking points in hope that their audience is brainless enough to believe that they're actually presenting an argument. It's just, it's laughable and it's sad at the same time. You can see how defeated they are, especially lately with the whole abiogenesis. I mean, James Tour's just demolishing, the origins of life. We had Dr. Carter, Dr. Hayes on last week, demolishing, a couple of their best papers. It's not looking good for him. Brother Jamie, I saw you were going to unmute, man. Go ahead.
I think a lot of what's going on, I've noticed, is that a lot of these people, at least the less sophisticated ones, they just accept that they know that we're all just making the stuff up as we go or something, and we don't have, they just assume it's all nonsense based on the Christian worldview, and they just, that's like the first thing that's going through their mind. So they think they can just, they don't even have to know the position really half the time. It seems like they're not even willing to engage the ideas, and
Right.
They can fall back on this notion that it's all lying for Jeebus or whatever,
Right.
And it's kind of sad, but it's kind of frustrating too. But it's so bad. It's like you almost feel sorry for them, really.
Yeah.
I mean, like I was talking to Amy Newman and Donald something. I forget what he goes by, Dak or something.
No, you're talking about Donald Kronos, one of
Yeah.
the noxious, loudmouth Hawaiian shirt wearing fools. Like he's the atheist version of Ken Hovind. Good.
Yeah, and that other guy that was like on Modern Day Debate, he was on there too one night, and like he's so overly confident, and like he doesn't even understand what atheism is, and he wants to like redefine it as being anti-theism as being the standard atheist thing, or lack theism, and doesn't even like understand it. But like that Donald guy, I think that was Randolph Richardson I'm talking about. He's a really nice guy, but he's got some weird ideas, and like they're so overly confident. But that Donald guy was like, he just always wants to say, like, for instance, like Kent Hovind is is totally like a scam artist, snake oil salesman, and it's just lying. Like he like literally on one side of his mouth he says he's not even he's like an idiot or doesn't understand what he's talking about, and then but most of the time he's saying he's like a total liar, and he knows he's teaching lies and he doesn't care, and he's doing it for the money. I think he really believes that, and it's amazing. But
have y'all seen the stream with Otangelo, Dr. Carter, Sal, and Donald?
Yes. Yeah.
Oh my goodness gracious! I was giving Dr. Carter a hard time. I was like, man, how did you manage to contain yourself with that guy?
With Donald interrupting every 3 seconds, and you can see him whenever someone's talking, he's not relaxing and waiting for his time to talk. He's right in there, just ready to interrupt. And he did the same thing with Ken Hovind. And even after James in the Modern Day Debate between them said, "Hey, listen, we're going to have to go into timed rebuttals if you don't stop interrupting." Didn't matter to him. Interrupt every 3 seconds. So Dr. Carter showed some great patience there.
Well, he did. And he told me after the fact when I asked him, I was like, how did you manage to not like lose it on that guy? Because I mean, Dr. Carter, obviously, first I was like, I was listening to him talk and was just like, this guy is a moron. And I was like, I can only imagine how much it must have been painful for you as a PhD biochemist to just be like, dude,
Right.
shut up. You are an idiot. because Dr. Carter was being very nice and kept like explaining why his standard atheist talking point was like not reasonable and not rational and like completely just annihilating. I was listening to Dr. Carter. Dr. Carter was like patiently just obliterating his stupidity. And then he would like spew the same talking point, like double and triple down on it. No, you just don't understand. And it's like Dr. Carter just broke down why your talking point has no plausibility on like 7 different levels.
Right.
And then you just told him he doesn't understand the chemistry. It's like, wow, man. Like,
A PhD chemist doesn't understand chemistry, apparently, according to Donald Kronos.
Dr. Dave!
Praise I am that I am.
What a train wreck. What's up, guys? Shalom, good evening.
I heard that you converted to evolutionism.
No, I was going to make a joke, but yeah, I don't think it's proper for the people here. The Garden of Eden.
Oh, yeah. We'll keep that off this channel. So, no,
especially at 3:30 a.m. your time, 2:30 my time. I mean, it's if we open that door, it's likely to degenerate into full scale, not suitable for children conversation. So
it's going to go from it's going to be a 3-hour night to about a 6-hour night that just descended into madness, devolved, which makes sense. We've been talking about genetic entropy. But yeah, what are your thoughts, Praise? You've been listening to some of the stream. And...
I have, yeah, and it's just a high level. Yeah, yep.
Boy, Praise. I was going to say this, John. And Speed of Sound, their little panel that they have when they try and rebut our videos and stuff. It's more like The View, if you're familiar with that show, than an actual intellectual response to our videos, because like you said, it's just complaining about our videos. It's also free advertisement for our channels too. I think that's why our subscribers keep going up and up and up, because all this free advertising from the atheists.
I mean, here's the thing though, I agree with you that happens to an extent, but very often they don't end up actually talking. Like they spew a bunch of like, standard talking point, blah, they don't know what to talk about, blah, it's so wrong, man, it's creationist, blah, that kind of stuff. And then they usually go off on like some entirely different topic, usually about their own like life problems. And like it turns out to like honestly, I think they're having like atheist anonymous meetings or something.
Yeah.
That's what it is. Last time I was there, they were like trying to read out of like a expired some old book about abiogenesis, and they were trying to get to the bottom of it, I think, and it wasn't going well.
You should have seen Speed of Sound attempt to rebut Dr. Jonathan Sarfati, PhD chemist, who we had on a few months ago. Time flies by, maybe even 4 months ago. And I made a clip, the end of abiogenesis, because within that 30-minute clip, Sarfati destroyed RNA world, destroyed carbohydrate first, destroyed all their best lines of evidence, right? And I think Speed got into like maybe a minute into it, and you could see it on his face where he knew that this wasn't going to be good for him. And he shut it off so quickly. And like John said, they just went into who knows what topic. But
So we're talking about this abiogen. Yeah. That guy doesn't know what he's talking about. It's totally. It's crazy. It's crazy. So the other day I was in at Home Depot, and
No, it's getting pretty bad. It's like Jamie was saying, they think we're spokespeople for intelligent design or creation, right? They say Ken Hovind's, he's a scam artist. So they really believe that, like you were talking about earlier, John, you were talking about alternative splicing, right? The fact that a single protein region, a protein coding region of DNA can give rise to how many? Hundreds even thousands sometimes of different functional proteins. Okay, so that's just one example. So they think we're spokespeople, we're scam artists because we're pointing out that, hey, listen, this, okay, did not come about by chance or non-design. You know, pointing out the obvious, right? But apparently we're just pushing some kind of scam for pointing out the obvious design in the genetic information systems that make life life. I mean, what's the thinking process? I don't get it.
Yeah, it's just bad thinking. Like the whole paradigm of evolution is like it's built up with all sorts of like, you know, just endless research done on trying to, you know, showing mechanisms of the way nature is. And now I'm not pretending like I'm really familiar with all the literature or anything. I just mean, if you just think about how they protect the premise so carefully, it's like, but yet they want to brag about how science is so open to being corrected, and you know, so long as it sticks with a certain assumption or whatever, and they don't separate, you know, what is fact from what is part of their, you know, interpretation or model. They can't even half the time recognize the difference. And I think that's really part of the issue. And it's like, what would it hurt to open up to like new concepts or whatever? Like they just assume that means it's gonna like hold them back or something, when there's just more and more evidence that that's not the case in any way. And hopefully that those kind of things will change and more people can be open or to realizing the reality.
Great points, brother. Great points. Yeah, it's yeah, go ahead, John.
Sorry, I'm trying to remember what I was gonna say. Yeah, I had a brain fart. Go ahead.
when you've been talking about this for 3 hours straight, you know, those moments become ever more common. Go ahead, Praise. What were you going to say?
So, what do you guys think of the Professor Dave's damage control? Like, did you see what he did the other? Like, I heard he responded back to Tour, and he was saying Tour is incompetent. And
really?
all these slanders. I just couldn't believe he would go down to that level. That was surprising. I mean, maybe not because he's an atheist. So yeah.
I didn't know he responded. I knew he was gonna, but I didn't know.
Yeah, I didn't know either.
It was a written one, as far as I know. I heard it was a written one, and he got butthurt on it. It was pretty bad.
His mom probably helped him write it.
Look, let's be honest. Dave recognizes that Dr. Tor is going to annihilate him. And he's got, you guys have heard me go on this tangent before, but he now has, okay, let's just think about this, right? You're a basketball player, okay? And let's say it's the 90s, and you're running around talking mad game on the street court. I've been like, look, bruh, I would whoop the ass. I would whip MJ's ass, man. Like, I would just take it. I'll be dunking on him all day, man. There's not a chance MJ can take me. Nah, man. You don't know what he's talking about, man. Like, fade away, psh, man, I got him all day. Jump? You think Michael can jump? You think Michael can jump, man? Michael ain't got nothing on my house, man. Right? And then through the grapevine, Michael hears, man, there's some little dude out on, a street court that's talking mad trash about you, man. Like, he said that you cost the team in 1988. It was your fault. Y'all lost in the playoffs, man. Like, you're the one that sucks. And okay, let's get real. Michael Jordan's gonna be like, yeah, okay, whatever. And then, oh boy, gets on ESPN and is like, man, I tell you what, Michael ain't got no game whatsoever. And then Michael's watching it after he's just, dunked on everybody, smoking a cigar, and he's like, man, this little punk be talking to, okay, you know what? I'm going to show up at his street court. I'm going to put him in his place. And then lo and behold, 4 months go by, little man forgot about it. And then one day MJ shows up at the court. And what's up, bro? heard you've been saying you got me, so let's do it. As the dude like looks up and realizes, oh man, MJ's 6'6" and I'm 5'5". I am toast right now. What happens? MJ like literally jumps over him, dunks on him. It's beat down. This is what is going through wannabe Dave's mind right now. The equivalent of the MJ has shown up in his court on the street, i.e., YouTube, and is like, "Boy, I am going to annihilate you." And that's what's happening right now. That's what is happening. And how does little man wannabe Dave make his money right now? It's doing stuff about science and projecting himself to the YouTube audience as being a quasi-expert in these different things. Now, MJ's coming along and being like, bro, you don't know jack squat.
Why 1988, John?
Huh?
Why not? Why in 1988? Was that Jordan's first year? Rookie year?
Nah, his rookie year was 1985, wasn't it?
Or 86 or something? I don't know. I got a rookie card, but
It was either 84 or 85. But I know I'm pretty sure they lost in 88 to the Pistons and then beat them the next year. But
Isaiah Thomas?
Lambeer, all those guys.
Yeah, Lambeer, yeah, all of them. But like the first year, like he missed shot, and the next year he hit the jumper on who's the white guy? It wasn't Lambeer, it was the other white guy.
Larry Bird.
No, Bird was with the Celtics. I'm talking about against the Pistons.
Dumars.
No, Dumars was black, man. The
it's the white guy. That's right, yeah.
It was the white guy, like at the buzzer. He catches like falling sideways, shoots it up at the buzzer, and wins.
No, that's the Cavaliers. You're getting it mixed up. Remember, he distorted himself in midair.
Dude, he contorted himself in many games.
It's Greg Elo, yeah.
It's Karl Malone. It's Karl Malone. Okay?
He's the mailman. Yeah.
Nobody got time for that.
I remember Larry Bird. He was the 3-point man. Nobody messed with Larry Bird.
He was the white wonder, the great white wonder.
Great White Hope. He hits him from the jumper from the right on the, what do you call it, out of bounds, you know?
I was actually a big fan of basketball in the Chicago Bulls days when it was like Tony Kukoc, Michael Jordan, Luke Longley, Scottie Pippen, Dennis Rodman. You know,
mid to late 90s.
Dream team. I wanted to be a professional basketball player back in those days. But then when Michael Jordan left, you know, all the fun ended for me.
Yeah, that was the end of the
Kobe Bryant. What are you talking about? I think Kobe's better than Jordan.
Not even close.
Get out of the stream.
Man, the Utah Jazz versus Chicago Bulls, Karl Malone versus
they beat the Seattle.
Wasn't it a couple years in a row too?
The Suns with Barkley and who was it? Barkley and
Dan Marley. Dan Marley.
Yep.
Dude, okay. Kobe, amazing. But Kobe Jordan was better than Kobe.
Oh,
you have to remember, Jordan took time off, man.
He went into baseball. He played for the Chicago White Sox. Remember?
Well, he never actually played for the Sox. He played for their minor league team, the Birmingham Barons. But
I thought he actually played for. Oh, really? Okay.
No, he never actually made it to the. No, he realized he sucked at baseball.
I thought he actually did. Yeah. Okay.
Yeah. He played minor. It was the White Sox minor league team is the Birmingham Barons.
Oh, okay.
Which he played for, but the point being, MJ, if he hadn't taken all that time off, would easily, because he took it off in his prime, he would have easily passed Kareem in points, because at that time, like, dude, he was scoring like 35 to almost 4,000 points a season, right? In that spurt, and he's like, yeah, I'm good. So, I mean, one and a half more seasons.
He just had style too.
At the rate he was doing it, he would have passed Kareem for all-time points.
He's a self-worshiper, though. Do you know what he calls his nickname?
Oh yeah, he's terrible.
He calls himself Yahweh. So he's putting himself on the level of God. So I'm just saying that's where it turns me off when Michael did that.
I didn't know he did that, but yeah, I mean, don't get me wrong, man. Kobe was amazing. Kobe was Kobe. It's a whole other.
Well, John, back to what you were saying earlier with your Michael Jordan. You got somebody who's talking smack on the street. That's how a lot of these guys are, like Professor Dave. It's a psychological game. Okay, so he recognizes that he's getting absolutely demolished. He's getting humiliated by Dr. James Tour. Okay, I mean, without even Dr. Tour, remember when we had Dr. Rob Stadler on, which Professor Dave was aware of. Dr. Rob Stadler demolished Professor Dave. So he's already been demolished. Right now he's getting demolished by Dr. James Tour. It's not looking good for him. So it's a psychological game for now him to, as Praise was saying, I guess it was a comment or something that he left. So it's his way of doing damage control. Still not tough in a comment section, even though he knows he's been defeated. In order that maybe a few of his fans or a few of his followers will see that, and they're blinded enough to think, oh, okay, we were worried, but our master, Professor Dave, responded. It's all psychological. It's like inspiring philosophy. He hasn't actually refuted Sarfati. He hasn't refuted Dr. Lyle. He hasn't refuted the arguments against his little video on young Earth creation there, but he has gone about and left comments on Facebook and stuff saying, oh, I watched, the response to my video and it wasn't very good. I mean, what kind of response is that? It's psychological, it's just give a response and hopefully some people will be blind enough to believe that
Well, and here's the other thing about Wannabe Dave. So you guys do recognize the Dr. Tour hasn't even gotten to the good stuff yet, right?
Yeah. So it's only getting worse for him.
So, I mean, the biggest stupidities of that video are yet to come. so when Dr. Tour is just getting started on the full scale, stupidity of that video. So, and we just had what, video 5? Guys, there's 13 of them. 13. So going back to the MJ analogy, right? So you've gotten dunked on, you've gotten schooled, you've gotten your ankles broken, you've gotten beaten down in the post. That was game 1, 2, 3, 4, 5, and then old boy's like, and MJ's like, no, ball up, bro. Next game, let's go. And you guys said, like, how many more do we got to do? And he's like, best of 13. And dude's like, I can't make it another 7 games. And then he starts screaming at everyone that's watching, like, man, this guy's being mean to me, man. He's being mean to me. Like, come on.
You're talking through a cup, LPP. Sounds like you're talking in a cup right now.
Is that an effect? You're losing you.
Think you're losing me?
Yeah, your voice is very faint.
Yeah, it sounds like you're talking through an old flip phone all of a sudden. But we can hear it.
Okay, how about now?
No.
Ah, crap it.
The cup just got bigger.
Hang on, I gotta refresh.
Maybe plug in, baby.
So, are you saying Dr. Dave is not the LeBron James? I use him as no. He's more like, high school wannabe, yeah.
JV. Hey, Standing. Have you? I was looking at I think it was an Andrew Snelling thing where he talks about radio halos. Have you seen that? Anybody? Are you Praise?
No.
Oh yeah, I watched it. Actually, I watched it today. I think he just came out with one on carbon 14. He talked about. It was funny because I did a video last night where I talked about radio halos, fission tracks, helium, as well as the so-called heat problem, carbon 14. And then he just put out a video, or I think it was Is Genesis History put out a video Yeah.
That's it. Yeah,
Yeah,
I was
that's a good
watching
one.
that. I was trying to figure out. I'm still a little unclear about the. I think I get where he's descending from Gentry, and I think he's probably right as far as, like he said in there about, well, there appears to be irrefutable evidence that some of the granite must have formed during the flood, or I don't know. It kind of seemed like it wasn't. He made it sound like it had to be the case, but because of the way it was part of the other sedimentary layers with fossils in them, the way it was sandwiched in there, so that the granite would have, some of that anyways would have to be have formed during the flood some way. But I'm a little unclear about the potential possibility of the ideas from hydroplate theory, and because they don't even seem to mention it, none of these other young earth creationists, about the formation of the isotopes or whatever. I just wonder though, where do they think God formed the earth with radioactive isotopes? I mean, because
So
they talk about accelerated decay, but sorry, real quick, just they talk about accelerated decay. Would have to be they basically say it have to be the case somehow, and they don't seem to ever mention the possibility that these heavy isotopes could be created from some process in the flood. So I wonder why they don't think that, or why they don't ever think that's a possibility or mention it. But then I wonder, where do they think these things came from? If they think God formed the earth like we do, and I just wonder about that, because that was part of the argument I think Brown has, and I think I've heard others use it too. It's like, well, why would God create it that way? You'd think it'd be more like those wouldn't be necessary or something. I don't know.
Yeah, that's a good question. Yeah, they don't really. You don't hear too much talk from, I guess, the CPT proponents on the creation of the heavier isotopes from, how we look to like the Z pinch and the electrical activity that would be going on in the rocks during the flood, during these catastrophic processes that would ultimately create those heavier elements. Unless they say through, I'd have to look it up on the spot and see how they, because obviously they're looking to accelerated nuclear decay. They've got their lines of evidence, that I think are strong when it comes to the radio halos and the fission tracks, like the scars directly in the rocks that, hey, listen, this has to be the result of accelerated decay, as well as the heat. So maybe all of that heat and energy created during the catastrophic movement of the plates, because you'd still have all that havoc being wreaked within the rocks themselves during CPT. Would they say that resulted in some of the heavier elements? Maybe they were there kind of dormant, because I can't see them being active during the unless they weren't there in the post or the pre-fall world. But when God removed his sustaining force, maybe they're okay in the pre-flood world with there being the heavier isotopes. Go ahead. I'm sorry.
Yeah, that's a good point. That's possible. I'd like to ask some Snelling or somebody else because he did point out that for instance, in the granite where these halos are found, the different versions of, but he was pointing out how, well, the issue is because of them having to be flood created granites. There's uranium. But he was pointing out how the uranium, which would be normally the mother isotope or whatever you want to call it, the first primary isotope before it breaks down. He said there's no doubt about it, based on today's half-lives, that at the same rate as we see today and measure it, it's 100 million years that would take for those decay, and there's halos of those too dispersed amongst the various daughter isotopes like radon and different versions of the polonium, like 218, 214, 210, and they're within like a millimeter of each other. and so I think that's why they insist that it would have to be the decay, because those rocks appear to be part of the sedimentary layers. But then there's another problem with the halos, like the one polonium like 214 or something, I believe, was like has a half-life of like less than a second. So like it's hard to explain no matter how you look at it. But I was just wondering, unless all these aren't just like a chain of decay particles, because some of them take much longer in between the stages. I think there's like 8 kind of just like whatever you want, stages, I guess, of the uranium. Is it lead's in there? But I just wonder, seems to me also, the other thing was that the fact that they still exist in the granite, like at least on the outer edges of it, because they drilled down into it, and he was saying as you get deeper, and they check the temperature of as you get deeper, like at 150 C, it erases the signature. Those halos are erased, or the rocks kind of like it just takes away that signature. So they start to fade and then disappear as you go deeper from like to 100. So it has to be like 100, less than 100 C or whatever to even persist in the granite. So it seems to me like what does that say about heat problems? Like the fact that they exist, period.
Yeah, I think the fact that these radio halos and the fission tracks too, the fact that as you go deeper into this drill hole, as the temperature increases, like you said, I think it's when you get to about 150 degrees Celsius, the radio halos themselves actually start to fade away to the point where they actually disappear, because the increased heat, it energizes the crystal to the point where those particles, apparently they repair themselves. So Dr. Snelling was pointing out the fact that, okay, if there really was as much heat given off as the critics say, then we shouldn't have the evidence from radio halos and from fission tracks. But yet we see the radio halos there. We see the fission tracks there, which indicate accelerated nuclear decay, which means there must have been rapid cooling during the flood, which would have mitigated the heat, right? So I think that's a good argument.
Yeah, how would the heavy isotopes on a molten Earth have done this, or whatever, in their model? So it seems problematic for any model, really. But to me, it also seems like it kind of points to me is this idea from the HPT model that I think I'd like to see like a layout of where he mentioned rare earth elements and stuff. I'd like to see where the concentrate, or how well they have that mapped out. Like, where are the concentrations of rare earth metals? Because I when I was looking at that briefly, I noticed that they mentioned something about meteorite water at these sites where the rare earth metals are, and it just jumped out at me. It sounded like, well, if hydroplate theory is right, that makes total sense, because that's theory is saying that certain kind of water, I forget what's in the water, but it's very specific signature, which can be created by the processes described in hydroplate theory, as it's shooting out along the edges. It seems like too much of a coincidence. I mean, I don't know how much of that theory, those ideas were developed based on such findings, but it seems like why would a meteor strike in these places where the Earth seems to be like showing these elements all along these like areas that are like large distances and stretches or whatever, along regions or whatever? Are they going to try? And then they find these meteor signatures of meteorite and the water that they find in meteorites at these sites. It just seems too hard to explain from a standard model anyway.
Yeah, I think these observations in the rocks, I think from a secular standpoint or a uniformitarian standpoint, I don't see really any real good reason for what we're seeing. And when it comes to the HPT model, I do like the way it explains the origin or the creation of the heavier isotopes. You know, and that's why I've had proponents of HPT on, obviously CPT as well. And I really like aspects of all the different models, you know. Even the VPT model from Mike Ord, the vertical plate tectonics, I think has a lot of explanatory power too. But I don't see this heat issue as really being a big issue, because we see, it's just like I was explaining to David in my debate too. I mean, we literally see the evidence for accelerated nuclear decay in the rocks, and yet that same evidence shouldn't be there if there was this massive problem that you're pointing to, you know. So
yeah, there's too many anomalies.
Yeah, that would so now we can sit here and hypothesize, okay, you know, all these different factors during the flood could have taken up a lot of the heat, you know, or we can look to plausible scenarios that could lead to rapid cooling during the flood. You know, we can come up with these plausible explanations because we know that the heat must have been, that heat disappeared somehow, was mitigated somehow, because if it wasn't, we wouldn't have the radio halos in the fishing track. So yeah, I don't see it as much of a, and I think we have a bunch of plausible reasons to believe there was some supercooling. I know, at least when it comes to the accelerated nuclear decay, when it comes to the creation of the heavier isotopes during the HPT model, I know there's what's that called? Cold repacking, I think. Where
yeah, from the fusion process, right? Like of the heavier, super heavy elements. And I want to understand more about how those break down, because I guess if they're like super heavy, like way above the periodic table, like, you know, when you start getting way up there and like over 200 or whatever, they're totally unstable. And so I want to learn more about how that could potentially, like if you had a bunch of just random super heavy elements being like formed like just for a moment or whatever because of the fusion potential fusion scenario, and that's called doing the cold packing and absorbing the energy that would be the heat, I'm pretty sure they would break apart into like, and they see this in the Z-pinch experiments, that like it literally like can form, like one strike of lightning essentially can like form every element on the periodic table in like a small quantities. And so if there was some high energy event, like who knows, like they were really big and they just broke apart into like every potentially random sized isotope on the normal heavy end of the periodic table, would that have everything you can imagine in between, you know, daughter isotopes, regular, you know, the parent isotope, and everything in between, and then they could all, I mean, would there still be an issue of needing so much time if like everything could be formed in that way? I'm not sure. I'm not being very articulate there, but
no, I think those are some good points. I think it's deserving of, you know, further study. Yeah, and that's what I like about all the different flood models is, you know, we see the evidence in geology for the Great Flood of Noah. And when it comes to the particular details, the mechanisms, you know, that's where these different models come about. And I think it's fun. I think that's kind of what it's all about.
Yeah, they all have useful data to show why our timeline of our ideas is not unreasonable because of the things that people point to that claim it's impossible. There's all sorts of data that says that's not the case, and so we can look at all the models and be happy that they're available to consider.
Exactly.
I'll shut up.
No, that's good. You're making good points. I'm looking at we're going on close to 3.5 hours. It's so funny how we could talk about these topics all day, because even when we went live, we're like, okay, we'll do about an hour and a half of genetic entropy. 3.5 hours later, we've touched on a number of amazing topics. So, but I'll say this, I got to get up pretty early, so I think I'm going to start winding it down and shut it down here. Jamie, brother, you're a wealth of knowledge. You should join more often. It's always a pleasure. Thanks for joining this stream.
Yeah, thanks, man.
No
Have a good
problem.
night.
Yeah, you too, brother. I appreciate this. Hope the audience had a good time. Hey, we still got 20 people in the chat, but SFT's starting to fall asleep. So I should probably call it quits here. Praise, thanks for joining for a bit as well. John just pointed out that he just saw the thumbnail now. Just saw the and that's too funny. It's what they do. They dodge, Neo Matrix style. So they dodge the data and the evidence. So I hope everybody had fun. Any final words, gentlemen?
I appreciate it,
God did it. All of it.
Yeah. Amen. Amen. All right, guys. Blessings and SFT and praise. Praise. You made it to the end, brother. You made it to the end. God bless.
I did.
God bless all.
God bless.